It's so good to see you together. So I hope you will enjoy the event. And as a start, I will ask our respected director to open this ceremony. So, director, please.
Dear colleagues, ladies and gentlemen, the National Laboratory is very young. 5 years ago, actually, a number of national laboratories sprang up in Hungary, and one of them was the National Laboratory, which is taking its anniversary today. I'm particularly grateful for those who traveled from abroad and visiting us, and it is very well known that our National Laboratory is keeping many connections abroad and within the country. And it is very important because we are living in a small, unfortunately, actually, all kinds of infectious agents are traveling very fast with us and causing several problems. And I still remember, and I was in the United States when the Surgeon General claimed that actually we are over with the infectious diseases. It happened in 1990, something like that. I may be wrong, maybe it was a little bit later. But we know that nature fights back, and new agents are jumping over species, barriers, and also the increasing resistance against antibiotics in the bacterial world is becoming a more and more serious problem. And that's why we are here, actually preventing the spread of diseases and taking care of diagnosis and therapy. And I'm quite happy that, definitely in Hungary, but I think that even within Europe, this national laboratory lived up to expectations. And as you will see from the lectures, it is really contributing very significantly to virology. And I really do hope that the National Laboratory, even if it is not going to be called a National Laboratory, is looking forward to a great future. So again, in the name of the University of Pécs, I welcome everybody, and please enjoy your stay here in the conference room and in the social programs. Thank you for your attention.
Thank you. So it's 5 years. 5 years since this building started to host a new institute. During the time when we started the National Laboratory, we have dealt with the global pandemic. So it was the establishment of the National Lab was a natural reaction to a challenge, which was a challenge of everybody in Earth. And then since then, several public health emergencies of international concerns passed. We dealt with them. And many outbreaks you never heard about, we dealt with those as well. Of the many lessons we have learned during this time, perhaps the most important is about the essence of science. And you may have a number of ideas for this. I have a good one. So what is science? I think science is finding the right questions and solving the right questions, both at the same time. I am proud that we have partners here, all of you, with whom we share common goals, shared goals to solve questions, and shared vision to find the right questions. We are very grateful to the rector for his continued support of our institute, and I thank to our partners present for helping us achieve this goal together, which is also the reason for the current event. We can now welcome you not just as a research group, but as an ever-growing institute. I wish everyone a pleasant time, and don't forget that this event is about celebration and networking, so I hope you will enjoy each other and find new connections, establish new connections to maybe solve the next question of science. Thank you very much. And right away, I will invite myself to open this section. And I will speak, it was very hard to summarize 5 years of a new institute. So I try to be focused on the history and the essence of our research. So our research and our interest for viruses started somewhere around 2007. And then we always dealt with questions with whatever we had. So that time we were very much interested in biosafety level 2 viruses, mostly hantaviruses, some other things, and we did whatever we could. And then step by step we built up a very complex system to understand these viruses as a complex system as well, because these are all of the outbreaks are not just simple outbreaks. They have ecological factors, they have molecular biological factors, they have virological factors. So step by step, we build up this academic research system. And since 2020, we started our institutional development, and thanks to this, we are here now as an institute. So where we position ourselves, the term pandemic sciences is a long-standing term, but it is recognized since the COVID-19 pandemic. So you can see, if you check the news, that several institutions are established based on this interdisciplinary field of science, which is focusing on understanding, detecting, preventing all these complex issues as we know outbreaks. So what we did in the last 5 years, we established 4 thematic research groups to cover the full needs of modern pandemic sciences and outbreak research. So you will see presentations from each of these research groups after me. And we didn't stop here. We also established the Science, Communication and Media working group, because especially the Hungarian colleagues may know us from the media. We are always all over the media, so this is crazy. It's like everybody wants the next pandemic to happen just now. So we always need to tell something which is like scientifically correct and we can calm people. So this is a responsibility, and that's why we established this working group. And also, we are lucky enough to have a university-owned company to help our partners from the industrial part, and also a researcher startup, thanks to our talented colleagues. So this is our system we work in. And what is the mission, and how can these research groups can be useful, because this is always a main question in modern scientific research. Okay, why is it useful? So the mission of these research groups, you can see like the disease vector and ecology, disease ecology research group, absolutely the global fight against arthropod-borne diseases. You hear a lot of these diseases, from dengue to chikungunya to European perspectives of West Nile virus, Crimean-Congo hemorrhagic fever. The list is very long. So this group is dealing with this problem. The Outbreak Prevention and Investigation Research Group, this is the Action Man and Woman Research Group. So they are developing outbreak prevention and intervention tools, research development with global focus. So they are absolutely working in the front line of outbreaks. We have the Pharmaceutical and Antiviral Research Group, General Drug Solutions against viruses. So we really target to find the general solution, not just to treat one virus, but to treat a whole family or more families of viruses at the same time. And then there is the Virus Host Interaction Research Group, which is the investigation research group to find the weak spots of viruses. And how can they be useful? Actually, these examples I list here, they already exist. So these research groups are doing all these activities already to different companies, to the city, to industrial partners. So I can say that these pillars of the National Lab really found their way. And this is the overall mission. So we discover new viruses or new challenges. We do research to find the solution against these challenges, and this is how we work. So our mission in one sentence is excellence and preparedness in the age of pandemics. And I'm happy to tell you that following the very basic concept of pandemic sciences, we build up our team based on these high standards. So I can say we are very interdisciplinary. You can see from biologists to hydrobiologists, communication specialists. We have all kinds of experts on board. And we manage people from high school up to PhD and beyond, which is very important in academy, and I believe also to develop the best experts for industry and academic places and everywhere. And also, which is I'm very proud of, we have a mentoring system with a background that meets modern labor market expectations. So we really take care of our students and colleagues. I will present you briefly our core capacities. Most of you are already familiar with these, and maybe it will be nothing new for you, but it's good to summarize them. So our core capacity is absolutely the Biosafety 4 laboratory. There are still not many of these infrastructures. And I really like this map because it shows you our responsibility. And this is how we think about our lab. We have a responsibility for the whole region. As you can see, we are totally the endpoint of available Biosafety 4 labs in Europe. And if you just watch around, just look around, you can see partners mostly from these regions we have already connection with, and we already have common goals and common questions to solve. The other very important capacity is our training space for biosecurity and biosafety. I can proudly announce that so far we trained more than 30 experts from 6 countries, which is, I think, a major leap in the internationalization of this. And we are currently developing the first standard training program with the help of our Brazilian colleagues, and also we play a key role in the ISO 35001 development, which is also a major leap forward. Our capacity, which came alive because of the COVID pandemic, is still active, and we are very proud to say, I'm very sad about these animals, but still more than 500 animals were used in different projects during the last 5 years. So you can see that it's a never-ending work, and this is also a very important capacity in a Biosafety 4 lab. There is a huge bottleneck worldwide for this. Another core capacity is our mobile lab. We are very proud to this mobile lab, and this is quite in alignment with our vision in South Asia and Balkan, 2 different but in many ways similar disease outbreak hotspots. We had several deployments in the last 5 years in these places. We developed a modular validated system, and we are in connection with the Hungarian company to develop the world's first absolutely standardized mobile laboratory system. So our strategy is in 3. The domestic strategy is being a virology knowledge center, a mentoring environment, and being ready for service anytime if anything similar happens like the COVID pandemic. We clearly have a regional role to serve the whole region, and also we have this idea to be the node to the EU, and we have already good results with this to help our Balkan colleagues and Balkan friends to be connected more strongly to the EU. And also we have a global vision to make partnership in pandemic sciences in places where it's most needed, finding synergies and working on the front line of outbreaks. I have an example for you. It's just a recent example. It was not originally planned in this presentation, but it just happened 2 days ago when the news came the WHO website that another Nipah infection happened in Bangladesh. And it's a fun fact, our team is already on site now since the beginning of January collecting these palm sap samples, because we know that most of the cases are connected to the consumption of raw palm sap. And you can see that this is an example that we are really on the front line of pandemics. We have a clear vision to build stable background in South Asia and the Balkans, and we have many different examples what we do here. We build capacity, we do teaching, we do research, and we make developments which are useful for the whole region. And I'm proud to say we have many, many different visions, and we already established local associations to help this partnership and to make more stronger background to our activities. I know this is always boring to see numbers, but I must show some numbers, and I'm very proud to present our digitalized background of the lab. So I'm also proud for this. This is the most important achievement we reached so far internally. So we made the whole institute fully digitalized, and it's super easy to show you how far we went in these 5 years. So more than 100 publications, mostly Q1, D1, a lot of partnerships, active partnerships throughout the globe. I am proud to have these, and this is the main takeaway message. So without you, this is not possible. So I hope to still stay with you in the next 5 years and build more numbers, solve more questions, and have more fun in science. Thank you very much. So I will ask the next presenter. Who is the next presenter? Sorry. Oh, yes, Tamás Görföl. Yes, sorry. I lost my schedule.
Yeah, thank you. I'm Tamás Görföl, and I would like to introduce the Outbreak Prevention and Investigation Research Group of the VNL, some results and some viewpoints. So some of the topics of this research group is coming from the earliest times of the virological research group of the university, and our main goal is to do effective outbreak prevention and control in the frame of the One Health concept. We are investigating different viruses, most of them zoonotic ones. We want to understand spillover, spillovers, and prepare for pandemics and other epidemics. So our main advantages are, of course, the BSL-4 laboratory. We have a strong ecological and taxonomical background in connection with the viral hosts, and we can do methodological development, and this also results in the making of the mobile laboratory capacity. The BSL-4 laboratory can allow us to investigate also human lethal viruses, and we can study, for example, filoviruses, paramyxoviruses, and this can allow us to investigate nearly anything. I'm basically a bat researcher. My studies were in bats and also viruses. So we can understand the host, the ecology and taxonomy of the host, and we can connect these two topics. So we can understand how to prepare for spillover events, how to hinder new epidemics. We have very good people in the group who can develop new methods, both in the field. For example, you can see a bat research equipment, which is a lightweight herb trap. We can sample bats which are the other groups cannot easily capture, and also in the lab, based mostly on nanopore technology, we can develop new methods to rapidly identify new viruses. Mobile laboratory is also important. We can identify a lot of different viruses on the field, and this means rapid outbreak response, and before spreading we can show that what is the unknown agent which is causing problems in the human population. Our main targets are high-risk pathogens. You will hear more about Yoviu virus, but also Nipah virus. Another important virus is canine distemper virus, but also vector-borne diseases like CCHF virus is also important. And of course, we are dealing with a whole bunch of different viral groups, of course, coronaviruses, paramyxoviruses, but we are preparing for diseases, and we want to find it before it will cause pandemics. Our flagship virus is Yoviu virus. It can cause mass die-offs in bat colonies. It is a relative of Ebola and Marburg, and it can be a model taxon for filoviruses. But Gábor will tell you more about this tomorrow. Nipah virus, mostly in Bangladesh, but we had some samples also from Vietnam, for example. We are using non-invasive method to study this virus, and we can isolate it. We also developed a new method to sequence the whole genome on the field, so we are preparing for this important virus. There were 3 field expeditions in South Asia, in Bangladesh, and we proved that we can sequence the whole genome on the site. Canine distemper virus is a widespread pathogen of carnivores. It can cause, beside the lethal infection of dogs, it can cause nature conservation problems in rare carnivores, also in Europe. Also, we can sequence the whole genome on the field, and we demonstrated this capacity in several places, in Bangladesh or in Cuba. So this is also a very new thing. CCHFV is getting more and more important. Together with Nipah, this virus is in the focus of the Sentinel project, which started recently. But with the Balkan friends, we already did some research in the frame of One Health in the past years. Viral discovery is also important. We study a wide range of hosts, including bats, rodents, carnivores, or even birds. We go to different field expeditions in Europe and in the tropics. And also we study the taxonomy and ecology of the hosts to prevent spillover. We use different methods, PCR, metagenomics, these are common. And we study nearly everything. Of course, metagenomics can find different kind of viral groups, but we focus on coronaviruses, paramyxoviruses, lyssaviruses. Not just the mammalian hosts are important, but the ectoparasites can be important. We also study ectoparasite virome, and we also want to reveal potential vectoral role of these ectoparasites, which can lead to spillover to different hosts. So this was my brief introduction to the research group. I hope the research will continue with more and more cooperation, and thank you for your attention.
Thank you, Tamás. So I will now ask Anett Kuczmok to present the antiviral research group.
Thank you. So it's a great pleasure and honor to welcome everybody and to introduce the work of the Drug Development and Active Ingredient Testing Group. The title of my presentation reflects a little journey. It started with the initial ideas and has led to measurable outcomes. And I will briefly outline what the main areas of expertise are and which milestones we consider main signification in the field of the active ingredient testing. I think everybody knows that the antivirals are a main subcategory of active pharmaceutical ingredients and play a main role combating viral infections. And these specialized design compounds designed to inhibit the viral infection and target various stages of the viral life cycle. And the development of these compounds requires extensive research and scientific expertise. So this is a long and complex journey which involving multiplied stages and an increasing number of stakeholders. And the main goal is throughout this process, prevention, a more effective treatment, and fewer side effects. Our team actively involved in the early phases of this development, focusing on in vitro ex vivo studies and sometimes in vivo preclinical studies. At the early beginning, we started the antiviral compound testing activities were launched with a small but highly motivated team. And as our research profile expanded and our research projects become more complex, the group grew and complemented and now creating a dynamic work environment. And we study and develop antiviral agents with a special emphasis on UV inactivation techniques and their biological applications, focus on vector-borne viruses, and focus on discovering suitable active ingredients for use in antiviral medication and marking the first step in the development of the antiviral drugs. We have approximately 500 novel newly synthesized compounds for the in vitro testing, and we can work several industry and company research companies in different topics. And we think that the reuse of existing drug treasures are the most important because they can immediately put into clinical trials if we have a problem. Our works always start with common and basic biological methods, and after we optimize every testing parameters and determine the cytotoxicity of the antiviral agents, we can evaluate the efficiency of them using reliability assays, molecular biology-based readouts, and immunofluorescence staining, and which allow for us to combine the inhibition at functional and biological levels. We started the active compounds testing with glycopeptide antibiotics and their derivatives in a cooperation two excellent chemists. They found that these compounds have antiviral properties against wide variety of viruses and synthesized teicoplanin, vancomycin, and rhizoxycin derivatives, and we examined them against, at first, West Nile virus. And we found that one extremely and fine moderately can block the replication, viral implication into the in vitro testing. During the COVID-19 pandemic, we also can work these compounds and the newly synthesized apocarotin derivatives. And it was very interesting. We found that the teicoplanin and rhizoxycin derivatives show a strong antiviral activation, and the apocarotins, the natural apocarotins, both of their own and incorporated teicoplanin form, has also showed the strong antiviral inhibition. In additional to them, there were several researches which resulted in international significant pharmaceutical research findings. After that, we were thinking about that, have antiviral properties of the glycopeptide antibiotics against adenoviruses, and the answer is yes. Because our results demonstrated that one investigated teicoplanin derivatives, the LA116, exhibited a broad spectrum antiviral activity. Its efficiency extended beyond to flavivirus to include alpha viruses as well as the coronavirus too. And it showed very low concentration effective values across a multiplied virus families and showed a favorable cytotoxic profile. So these findings highlighted that the potential of glycopeptide antibiotics and as well as a novel class of antiviral agents, and had led to filling and patient application covering the antiviral use. During our works, we have opportunity to work nucleoside analogs and their derivatives. These compounds also showed inhibitory activation, not only against the coronavirus, also alpha viruses too. And based on these results, we are currently preparing an additional patient application. Also, we can identify several promising vancomycin agricon derivatives, which show inhibitory effect in vitro West Nile virus and the Crimean Congo hemorrhagic fever. And furthermore, in the framework of Eduquite Seed project, we can collaborate with young postdoctoral researchers and investigated compounds synthesized by the Prague Institute of Organic Chemistry and Biochemistry. We also identified promising candidates against the CCFG virus, but unfortunately I cannot share even more details because these projects are ongoing and currently confidential. But we incorporated human cell lines in our studies, partially focusing on the infection-induced gene expression changes and cell response which triggered by the viral infection and antiviral treatment. And at first we used a neuroblastoma cell line infected with Symbis virus, and we observed that the infection triggered caspase-dependent apoptosis and triggered robust transcriptional upregulation in immune innate genes. So this result elucidated the temporal dynamic of Symbis virus, and we get better understanding the Symbis virus associated neuropathogenesis. And in the final, we utilized a special enclosed system designed by the Rolling Biotechnology company to allow selective exposure of the Zika virus to a UVC light. This irradiation, 45 seconds, effectively inactivated the virus with no cytopathic effects, no significant changes in the quantification values, and its preservation the whole viral genome. And microscopy pictures demonstrated that the virus can enter in the cells but didn't replicate. So this application can make safer and user like for vaccine development or diagnostic access and or antiviral research. So in the next and now, we are working on mapping the viral inhibitory mechanisms of patient-pending and promising agencies, including the work additional human cell lines, new virus, new substances, and testing the resistance. And the future research is focused on the additional application of the UA inactivation, and further investigation the immunological and epigenetic processes. Beyond the day-by-day research, we were very active internationally, so presenting our results, different conferences, and have opportunities to visiting the collaborating partners' laboratories, and our young colleagues, researchers. They are very active and engaging communication our research, and in different public events, different media platforms, and they are very active in education and training processes. And Yes, in the final, I would like to say many thanks to the team, their collaboration, and to you for your attention.
Thank you, Annette. I will now ask Safia Zegbik to present the virus-host interaction research group. Thank you, Safi.
Hello, everyone, and thanks for being here today to celebrate our 5th anniversary. 5 years of challenges and successes as well, and as a proud member of the VNL, I will introduce to you the virus-host interaction research group. We are a multidisciplinary team of 6 members. We range from master students to PhD researchers, and we aim to understand the virus-host interaction from several angles. And within the VNL, our rule is to deliver the mechanistic foundations in order to help both viral discovery and antiviral strategies. Our main interest is zoonotic viruses, and now on the focus are hantaviruses, coronaviruses, and Nipah viruses. However, we are also investigating some anthropootic viruses like hepatitis C. We aim to understand the emergence, adaptation, the host response, and the effect of interventions on this interaction. Okay, so we act as a gateway between Europe and Africa, and in order to support the global health, we established collaboration in Algeria, Tunisia, and Morocco. So what we do and how we do it, so basically we use in vitro, in vivo, and in silico approaches in order to study the interaction on different levels. On the cellular level, to depict, for instance, the viral tropism on different levels, or to study immune responses as well. We shift toward the molecular part, and we implement studies on expression or viral detection as well. On the evolutionary, we aim to understand the co-evolution of the virus and its host, or also to predict some, for instance, to predict antiviral resistance. Some of our key findings and methodological development of hantavirus isolation, which was presented in different conferences and by our student Greta Verga, who earned the 3rd place in the National Scientific Student Association conference. We also work in collaboration with pharma companies like the Rolink Biotechnology, and there is already an ongoing project on hantaviruses. Some of the key findings is the detection or the evidence of expansion, host expansion in the case of shrew-borne hantaviruses. We also investigate the effect of essential oils as potential antivirals, which are a soft alternative to synthesized drugs. No offense, Annette. So in the collaboration with the Department of Pediatrics, we assessed the magnitude and the quality of the antibodies generated upon a COVID-19 vaccination in pediatric oncology patients versus healthy patients, and manuscript is already under process. And in collaboration with the Faculty of Informatics at Alta University, a mathematical model of hantavirus infection spread is under development. This work was presented already in a conference. On the international level, our student, Fatima, performed the first evolutionary analysis of the SARS coronavirus 2 in Algeria in collaboration with Pasteur Institute. She was able to detect some super spreading events and some Algerian characteristic mutations. Okay, so we move forward with Nipah virus, and we try to depict the regulatory network between the mRNA are involved in. And for future perspectives, we aim to expand the collaboration network mainly in the MENA region. We would like to test in more advanced systems like human organoids, to investigate at the single cell level, and also to translate our findings by developing tests and help in antiviral drug development. Also, we would like to develop a clinical virology unit. Thank you for your attention.
Thank you, Safia. So the next round is on me. Cornelia is giving a keynote speech today in Trento, so I am proudly presenting the vector disease, the ecology of vector-borne diseases research group. Disease vectors and disease ecology research group, sorry, too much ecology. But we are very proud of this research group because I believe that the most complex system of infectious diseases is lying in the vector-borne diseases. So this is the mostly affected infectious disease type by ecological backgrounds, climate, complex social networks, a lot of factors which are clearly giving a challenge to understand these. And here are some examples known or maybe not known for you, but for me, like I always tell this to my students in virology, that the urbanization of yellow fever virus in Brazil, for example, this is clearly because of human activities. But there are a number of other examples, like the spreading of Asian tiger mosquito, the spreading of the hyaloma ticks, which are the main vectors of Crimean-Congo hemorrhagic fever virus. So clearly there are challenges, not just in tropical regions, but also in Hungary with vector-borne diseases, which we must understand soon. So what is the relevance of vector-borne disease research? You clearly have some ideas for this, but this is a good example for the complexity of research we do. So the background in ecology, virology, and biotechnology is absolutely necessary to understand and fight these diseases. The experience in mosquito monitoring and research is also a must, and we are lucky to have this expertise in this research group because of our long-standing history in this research. The support of mosquito control is a must. You may have heard in the news, in the Hungarian news, that year by year we have this problem with mosquitoes, and we must deal with them in a more complex manner than we do now. Also, we are helping local, the city here and other cities in Hungary to deal with this challenge. We have experience with several mosquito, tick-borne, and other vector-borne pathogens like West Nile, Sinbis, dengue, Zika, chikungunya, and a lot of others. And I'm proud to say, but thanks to the ongoing research we do now for more than a decade actually, we have a mosquito-specific virus bank in our freezer, which was isolated from local mosquitoes and which gives us a stable basis for the future perspective of research we plan now. And also we are engaged because of these capacities in several international research networks. And just to name some, here is the AIM-COST action, which benefited us with we are proud to have a couple of you from this network now here. And the other one is the Balkan Association for Vector-Borne Diseases. This is a recently established association, but very useful for the whole region, the VectorAct project we have. And also in parallel to this, and following the basic concept of our institute, this research group runs science communication projects as well in relation to vector-borne diseases. So what is the mission here is, in one word, know your enemy. But how can you know your enemy? Of course, if the challenge is so complex, You need complex manners to understand this and fight these challenges. So we do distribution mapping, dynamic analysis, environmental factor measurement. We try to understand the preferences and the needs of these vectors, like the habitat, the feeding activity, the breeding season. We try to understand the relations between populations, phylogeography, the basic genetic background which shapes the actual pattern of these vectors in different places. And the most important, this is the crown jewel of vector-borne disease research, is understanding the vector role of different species. This is a very complex research, and we also have some activities in this. And I will present in a different presentation, because this research group is now building a new laboratory capacity here in the institute, so I will speak about that later. But that will be a major help in understanding the vector role of different species here. And of course, we must do, this is a must do, and I think this is the most useful for everybody sitting here, is understanding the efficacy of the current control methods and helping with scientific data, actual scientific evidence, where to go and what to do as a next step. Here are some examples which we are deeply engaged in. One very important topic is understanding the distribution of hyalomatics in Hungary. This is a very important vector because this is the known vector of Crimean-Congo hemorrhagic fever virus, which is a very important virus for the whole region. And just a couple of years ago, we established, this was a new idea by the Ecological Research Center, why not to ask citizens to send tick samples. And this turned out to be very useful, so we received a lot of ticks, and unfortunately many of these ticks are hyalomatics. So during the last couple of years, we were able to identify continuous hotspots of the tick, and they really don't care about our winters. So they are there all around the year. So I can tell you now that we have stable populations of hyalomatics, so it's just one step ahead to have the virus itself. Another very important example is the different invasive mosquito species. They are not just nuisance for people, they are not just pissing you off during your barbecue party in the summer, but they are very important vectors of different diseases. And this is what Cornelia is doing now, the workshop. So she is summarizing the knowledge about the Aedes coraicus mosquito in Trento, because this is one of the invasive species. We have several invasive mosquito species in Europe. And the flagship mosquito species is the Asian tiger, of course, because it's a well-known vector for different diseases. But there are others in the background, and there are not much scientific focus on these vectors. But anyway, they are spreading like wildfire. And the greatest example for this spreading is the Aedes coraicus. So we do a lot of stuff with Aedes coraicus, from the basic genetics to population dynamics to establishing complete genomes to give the tools for future perspectives and future research to better understand this vector. And I can tell you as a local, I don't know, famous thing that Pécs was the first city where it seems, as we understand now, Aedes coraicus emerged. So we had this duty to understand this vector. We have this special role here in Pécs. And of course, we are developing tools. This is a must to do. So we have a couple of projects to use our mobile lab concept to help control companies in Europe to better control mosquitoes. Or we are also developing cheaper and better surveillance systems to understand disease dynamics. And here are a couple of examples. We are really testing the feather of birds. We are using different sources of secondary samples from hunters and other sources, and we are trying to establish stable connections with government organizations, private companies, and whoever is needed on board to understand more rapidly these challenges and these diseases. Also, we try to react. Like when anything happens, like this is a story about Pécs again. So we started to have human cases of Dirophilaria recently in the last couple of years. And the local doctors contacted us, guys, what is going on? You must have some IDs. And then we had some IDs, but of course we had to test these IDs. So during this last 2 years, these IDs and questions turned out to result in useful data, especially regarding invasive species, local mosquitoes, and the environment itself. So now we have a clear picture about the very complex and diverse connection network of the mosquitoes, the hosts, humans and the potential spillover possibilities of different pathogens. So this is a great example about what we do. Also, we started to open the Caesar, and we are now trying to understand not just the ecological complexity of these vectors, but also the microbial complexity of these vectors. And when we make one step ahead, we see like 5 more questions popping up. So now we have a major question like solving the diverse and dynamic microbiome, the questions in relation to the microbiome of the vector itself, in relation to different environmental and non-environmental factors like microplastic pollution, the temperature, environmental conditions, all the things. And this is another reason why we started to establish a new lab, to be able to model these complex scenarios and involve as many factors as we can to understand these complexities. So this was the presentation of the vector group in short. Thank you very much for your attention. And I'm sure many of you are hungry, just like me, so I will ask now Balázs Somogyi to give some ideas about the exhibition we have. I'm very happy about this exhibition, by the way. So please, Balázs, the stage is yours.
Thank you. I have the honor to open the exhibition of the photos, but actually the photos are not here, but let's give it a try. So 5 years, more than 40 persons, and 30 pictures. 30 nice photos. This is our history, but of course it's just a part of our history. But behind every photos, every pictures, there are some personal stories too. Sometimes these stories are happy and nice, but there are some stories behind us, what is hard or sad stories. Just let me talk about 2 pictures, because in the morning when I watched these pictures again, I stopped next to the or front of the photo named Helping Hand. And I've just discussed with Dr. Zona that was a hard time for us, because we worked day by day, all day on the week, almost 20 hours on a day, because we had a part of the COVID diagnostics. And we've been exhausted, we've been sometimes depressed, and so on. So it was a hard time. But one day, a small group of photographers arrived, and they said that, hey guys, we want to take some photos about you, because we want to show the world that what are you doing here. And in the beginning we said that, okay guys, if it's really necessary, you can do it, but who cares about us? And after the first series of photos, they said that, hey guys, now we understand what are you doing here, and we can really say that you are not just workers in the lab or working on COVID samples, but you are the true heroes. And after that, we started to think in another way, that okay, for every day it's something monotonic and it's something hard for us, but we can do it for another aim. And to talk about some happier stories, there is a picture, it's called Emergency. And because we have a BSL-4 suit stuffed with some papers, and usually we use it for training. And of course it's faced with my picture. And one day I started to play the American wrestling entertainment something, so I fought with myself actually. And when I slapped it slapped me back. So somehow it became some schizophrenic something, like in the Fight Club movie, because I started to fight with myself. So I highly recommend to you that let's find us, all of us from the National Laboratory of Virology, and let's find together the personal stories behind every picture and more. So thank you.
Enjoy the lunch and the Thank you. Thank you.
Megy? Okay. Hello and welcome everybody. I'm Daniel Hanna from VNL and Rolling Biotechnology. And today I have the privilege to, please put up my presentation, to talk about Hungary's PhD excellence frameworks. Just one moment, please. Okay, until they fix my slides, I will just briefly introduce the topics I will be touching today. I will talk about the who, the what, the do's and don'ts regarding Hungarian research programs, government policies, narratives, and institutional PhD excellence frameworks. A brief rundown of what I will be talking about today. First is the national strategic priorities, namely the Neumann János program and the Hungarian smart specialization strategy, around which Hungarian grants and funding are revolving. After that, I will talk about VNL's institutional framework and then the industrial partner, namely Rolling. And after that, I would like to introduce all our excellent PhD students and the awards they won, or like the program they are involved in. So let's start with Neumann János program. This program was introduced by the, back in the day it was called the Ministry of Innovation and Technology. Today it's called the Ministry of Culture and Technology. It was introduced in 2023, and this basically outlines the how of research and development. It emphasizes or like puts into focus the academy and industrial connections, relations, and its long-term ambition is to put Hungary in the top 10 global innovators by 2040. Neumann János program revolves around three core concepts. One is associate, meaning it wants to foster international relationships with academic and industrial partners. It also wants impact, be it economical, societal, or whatever, and it wants measurable outcomes. So if you submit a grant today in Hungary, these are the three key concepts that you have to outline in your grant application. The smart specialization strategy, on the other hand, emphasizes on the what. It basically gives you the areas on which you should focus and for which the Hungarian government would gladly give you grants. I will not read you all the information there, but the slides will be provided to everybody with links underneath. So, why is it relevant in our context? Because basically, VNL embodies everything that VNL and Rolling's cooperation embodies everything that the Hungarian government asks for. If you look at the smart specialization strategies, these pictograms are kindly provided to us by the ministry. We touch subjects such as health, services, digitalization, and cutting-edge technologies, and also the horizontal priorities like training and education, and the public sector and university innovation. Let's talk a bit about Rolling and VNL. We are situated here in our mignonette-scented place, Hungary. It's a flower, it has a very floral and sweet scent, and it's a common phrase used in Hungarian to describe Hungary. And we are in a very special place because we have a very big research infrastructure concentrated in a small place with all the necessities like the National Blood Transfusion Service and the whole clinical complex to foster research. And we also have everything in-house. with a BSL-4 facility with all the associated capacities. Rolling, on the other hand, provides an industrial framework with a slightly different mindset, which we will touch later. This is the team of VNL. It's ever growing, so this picture is not up to date. We are, I think, 50 and counting, or more people. We are very happy to welcome new students every year who are actually more and more excellent, which endangers my job. So let's look at what VNL does. I included pictograms so everybody knows but nobody understands. But I arranged everything. Basically, our core is, or like the core alignment is the health and medical relevance. I think it's pretty clear how virology and epidemiology is connected with health and medical relevance. The other big part is biosafety and biosecurity, which is in its infancy, not just in Hungary, in the world. The other, which is a new wave, so to say, in VNL, is a service-oriented mindset, or like we try with our certifications, like ISO certifications, to move into a direction where we can offer viable services, not just for Hungarian partners, but for every partner worldwide. Our research has always been collaborative driven. I think VNL has a reach in the whole world. I don't think I can name a country in which my bosses cannot give me a name, an address, and a phone number, who will answer actually. Other than that, the education and training capacity is still underutilized in VNL with 50 people. Our outreach and dissemination is also ever growing, with emphasis on our social media appearance. We have a dedicated personnel for that, and she does a pretty good job, or at least better than I could. We have state-of-the-art high-value infrastructure, that's a given, and we also have highly digitalized operation, which is also a recent addition to our roster. The ISO certificated operational framework has been already discussed. So, VNL represents academic excellence, true, but even that needs a heart. This is where rolling comes in. So, what can a company give to a big university that it cannot accomplish alone? The main 3 points is the translational mindset. It's just a different ballgame altogether. State-of-the-art infrastructure, that's also true for VNL, but not in a speedy and executive-friendly way. Also, we have direct market and partner access, which sometimes comes in clutch in our projects with VNL. So how does it connect everything in the cooperative doctoral program? In Hungary, our government wants to make us produce students, or like after that workers, who are well-versed in the academic parts and also the industrial processes, and who are capable both in the academic and in the market-driven sense. So let's look at the KDP program. It's very high-end, very prestigious. As you can see, it's roughly 10,000. We have in Hungary roughly 10,000 PhD students. The ECUP, which is the standard grant the Hungarian government gives, is awarded to roughly 500 students, which is roughly 5%. And the KDP, the maximum is 150 students, but the government doesn't give out the maximum. They even explicitly said that if they don't get good enough grant applications, they will just give out 0. So in this year, I'm sorry, just the dates are a bit off because I go by the official data and we don't have available official data for every year, but this is the latest we can have. So in 2025, the ECUP KDP was awarded to 119 students, out of which 2 are here in Pécs with us. In regards to the cooperative doctoral program, the expectations are basically copy-pasted Neumann János program and S3 strategy. It requires academic excellence and requires involvement in basically a university-industry cooperation. It has a high emphasis on product development, going to market, and other activities which are rarely associated with academic life. So, our track record. This is for 2025. We have a 100% acceptance rate, which is outstanding. 2 out of our 2 students are accepted into the KDP program. We have increased to 300% because we already had 1 KDP enrolled student last year, and we increased it to 300%. This is a good number, I just included that because it looks good. And other thing is, because VNL is judged based on our contribution compared to the whole university. The whole university, PTE, won 8 places in the KDP program. Out of that, 25% is here in VNL, which takes up, what, roughly 1 to 5% of the whole university student board. So we are small, but we make an impact. So who we are proud of? Please give a round of applause to all our winner KDP students, Vivian Kish, Greta Varga, and Peter Szabó, who I think are with us. I don't point to them because it's a laser, but they are sitting there. Please stand up. You are the pride of our VNL. Please stand up. And also I would like to thank their mentors for helping their success and for aiding them through their journey of becoming a PhD student. The mentors, I will not name them because I tried to keep it short. So basically that's all. Today I have one more duty. I will be the one to call for the presenters. And every time the one presenter is done, I will ask for the next one. So please welcome Professor Anikó Borvás from the University of Debrecen.
Thank you. It is a great pleasure and honor for me to be here and celebrate together with you the 5th anniversary of the National Laboratory of Virology. I came from Debrecen, University of Debrecen, and we are organic chemists at the Faculty of Pharmacy, and we are with a strong collaborative work together with the antiviral group headed by Annette Kuczmok. We provided the compounds for them, and they performed the antiviral studies. This story dates back 7 years, and it started with the antiviral study of glycopeptide antibiotics. As Annette mentioned this morning, the first compounds were glycopeptide antibiotics, and the activity of these compounds was first tested against West Nile viruses, and then this antiviral testing moved to SARS coronavirus when the COVID emerged. And then it continued with the antiviral testing of these glycopeptide antibiotics against different arthropod-borne viruses. Regarding chemistry, we modify native glycopeptide antibiotics, and from chemical point of view, it practically means lipophilic modifications, because or dually hydrophobic and lipophobic modifications, because it is generally necessary for the antiviral activity of the compounds. But we also produce in Debrecen nucleoside analogs. In the field of nucleosides, we are dealing with two types of synthetic modifications. One of this approach is the so-called thiol-in chemistry, which means photo-initiated thiol addition onto a double bond. Practically, it means that we introduce some exocyclic double bond to the ribose unit of the nucleosides. And then these unsaturated nucleoside analogs are subjected to thiolin reactions under UV irradiation. And the compounds are quite interesting because these are nucleoside analogs with non-natural configuration. It means that depending on the position of the substitution, the compounds have the xylo, the arabino, or L-like configuration. Some of the compounds were tested against different viruses here in Pécs and also in Prague in collaboration with another virology group headed by Jan Weber. And the compounds showed quite potent activity against chikungunya, Sinbis, and viruses and SARS-CoV-2 viruses. And we could establish some nice structure-activity relationship in this field, and the work was published last year. But today I want to talk about a bit more detailed with another type of nucleoside analogs. These are the morpholinonucleosides. Morpholinonucleosides are well-known compounds in antisense field of nucleic acid research because phosphorodiamidate morpholino oligonucleotides are approved drugs in the treatment of Duchenne muscular dystrophy. So the synthesis of morpholinonucleosides is well established. In this field, we wanted to prepare something new, namely tricyclic and bicyclic nucleoside analogs. The most interesting structures in this field are the bicyclic compounds. Why? Because bicyclic nucleoside analogs are very promising also as monomers or monomeric building blocks of oligonucleotides, because in oligonucleotides the bridged or locked nucleosides confer some kind of rigidity, which can lead to very high duplex stability. So these compounds are well studied, and other type of bicyclic compounds showed nice anti-tumor or antiviral activities. So we decided to produce an absolutely new type of bicyclic compounds, starting from not native ribose containing nucleosides, but morpholin containing nucleosides. So we wanted to produce compounds in which the morpholin ring is condensed with some other heterocyclic rings. Our idea was to use the starting nucleoside analog, which is nucleoside secodialdehyde from the nucleoside side, and some difunctional amines. And our idea was to produce bicyclic compounds using reductive conditions. It probably looks very simple modification, but it is a very challenging from synthetic point of view, because the intermediate is a hemiaminal, and it requires to subject reductive conditions to produce the expected bicyclic compounds. And what is the major question in this synthesis is whether the second cycle can survive the reductive conditions, or it also suffers reduction, which then lead to monocyclic morpholino, which is not so interesting. And the other questions are the level of regio and stereoselectivity, which can be achieved during the synthesis, because two regio isomeric bicyclic structures can be formed, and as a new chiral center is generated in the synthesis, many stereoisomers can also form. So I don't want to be boring, so I don't want to go into the details, but we performed a thorough optimization, and the conclusion of this optimization was that if the amines, difunctional amines contains thiol or a second amine function, then we can exclude the full reduction, which produces the unwanted monocyclic morpholino, and we can produce the desired bicyclic compounds. The ratio of the regioisomers mainly was governed by the type of amine reagents, but we can exclude the formation of the monocyclic compounds, and the reaction proceeded with the high or complete stereoselectivity. However, when we used amino alcohols as bifunctional amines, only the monocyclic morpholino compounds could be produced. I just show you some example, so representatives of the compound series which we produced, and these compounds were synthesized from uridine and adenosine, and the whole series were tested in Pécs and also in Prague. Currently, I cannot tell you about the results obtained in Pécs, not because the results were so poor, but in contrary, the compounds proved to be very potent antivirals, and we wanted to submit patent application in this field. So I ask you to forget about these structures because it will be patented soon. But I can show you some anti-SARS-CoV activity results. These antiviral tests were performed in Prague, as I have mentioned, in collaboration with Jan Weber. And the interesting point of the antiviral results is, at least one of the interesting points, is that none of the adenosine derivatives showed any activity against SARS coronavirus. and 5 compounds showed antiviral activity in human collu cells. And these compounds were selected to determine EC50 values, and only 2 compounds showed good activity with low micromolar EC50 values. And we wanted to elaborate the mechanism behind this antiviral action. First, we performed some in silico study by choosing several potential protein targets. The main protease, NSP14 and NSP15, were the compounds which showed some good binding with the compounds. And then we searched for some collaborators to perform enzyme inhibitory tests. Unfortunately, in these tests, we got only negative results, so none of the compounds showed enzyme inhibitory activity against these proteins. And this is the results which are the latest results in this field. So we, or our collaborators, performed some cell-based assay to elaborate whether which is the phases of infection when our compounds showed activity. These tests were performed in 2 cell lines, Viro, which is a green monkey kidney cell, and human lung epithelial cells, collu. These are the most common cell lines in anti-SARS-CoV inhibitory studies. But is interesting, the compounds showed higher activity in the human cells, and in all tested phases of the infection, the compounds showed high activity. Less potent activity were detected in viral cells. So we know that the enter of the cells is different in the 2 cell types. In Viro, the viruses enter by through endosomes, and in the case of collu, a direct membrane fusion occurs. So this is one of the point, one of the difference, and we will continue this investigation to elaborate the exact mechanism of action of the compounds. And as I have mentioned, the test of the compounds in PH is also in progress. And at the end, I would like to thank our colleagues and collaborators who participate in this work, and thank you very much for your kind attention.
Thank you, Professor, for the presentation. I would like to ask Dr. Geiger Röst from the National Laboratory for Health Security from the University of Szeged.
Thank you very much. First of all, I'd like to thank Gabor for the opportunity that I can talk here today. I'm from the National Laboratory for Health Security, which concerns with health conditions of a population of Hungary. So, and I'm particularly work on modeling of epidemic outbreaks on a population level. Today I will try to talk a little bit about virological problems and how we can utilize such methods possibly in virology. So my view of virology is probably very naive as a mathematician, so please forgive me if I'm going to say something silly. So, but in my naive way, I imagine virology experiments as the following. So I want to do some experiments with maybe 4 different strains of a given virus. And I want to use 5 different initial amounts of the virus in my experiment. And maybe I want to try 6 different antivirals and see how they perform. And I want to use in the experiment 3 different initial concentrations of the antivirals. And maybe I want to use 2 different cell lines. And I want to test 3 different treatment regimes. Maybe start the treatment very early, a little late, or much later. So now, it's just now elementary school level of mathematics that if I want to do all this, I need 2,160 experiments. So you can see if you have so many factors to consider, then the number of experiments, if you want to explore all of the possibilities, is growing extremely fast. So what if there would be a possibility that we just do some of these experiments, maybe some combination of the ones I mentioned, and we could predict the outcome of all the rest, and we could save a lot of time and energy and money and efforts. So this is what in silico viral dynamics can possibly be useful for. So in silico just means in the computer, that's the in silico. So we run these experiments in the computer, and we use mathematical or a computational representation of the viral dynamics. So they can be formulated as mathematical equations or a computer code, but the goal is to represent the elements of a virology experiment and the processes that are ongoing in the experiment. So the models have similar components as the experiment has, populations of cells, target cells, infected cells. Can have virus, antivirals, or whatever is important in the experiment. And then, of course, we want to calibrate these models, set the parameters that they give similar outcomes that we see in the experiment, and try to validate them using experimental data. So this is the classical example, a basic model of viral dynamics. So still, you don't have to be afraid of the equations. They are very simple equations. It's still undergraduate level mathematics. And basically, what the equations tell us is just this very basic process that you have an infected cell. It's on the blackboard figure. Infected cells produce virus. The virus can infect other cells which were uninfected before, and the infected virus will die at some point. So these processes are represented in these equations with parameters such as virus production rate and so on. So if I solve these equations, I can find these curves, solution curves that you can see. One figure is for the cell populations, other is for the virus concentration, and you can see these curves are very similar as you see in epidemic outbreaks. It looks like epidemic curves, very similar. Virus concentration goes up, it has a peak, and going down. So does this simple model ever work? And sometimes even these simple models can be useful. Here's one example I chose. It's not our work, it's a classical paper from the literature. It's about influenza, and what you can see in the figure is you can see the measurements, viral titer of a patient, and you can see these black curves, which are the outcomes of these equations that I just showed you in the previous slide. And you can see, you can get a reasonably good fit. So I point out that this is on a logarithmic scale. So you basically you see almost 2 straight lines. Straight lines on a logarithmic scale is exponential growth or exponential decay in reality. So basically what we see, exponential expansion phase, and then exponential decay phase, and there is some transient time in between when exponentials are not a good descriptors. But basically it's as simple as that. So besides fitting these curves, model outputs to real data, what was the use of this model? So in this paper, models revealed a number of things. For example, they could estimate the eclipse phase as 6 hours. Eclipse phase is time from infection of a cell until the time it starts to produce virus. they could estimate infected cell lifespan as 11 hours. And they could also estimate that from a single infected cell, how many secondary infected cells we can expect, estimated as 22. So there are some quantities here which are clearly important, but very difficult or impossible to directly measure. But through fitting such a model and finding the parameters, we can estimate them. So this model doesn't say anything about space, but we're also interested about space, where the cells, infected cells are in space and where the virus is in space. So we have to expand these models to spatial models. And that's what we did with our colleagues. We used a hybrid approach. So if you consider space, then you have size also. So you have very different size from virus and cells and molecules of the antivirals and so on. So that's why we use the hybrid approach. So basically, we model cells as individual entities, but viruses or antivirals we model by concentrations, and we use diffusion about how they spread, and cells can, of course, do a number of things, move on a lattice, and so on. So this setup is just a two-dimensional in space. You can imagine these layers on top of each other, just for the modeling purposes, and when on each layer we have one component of the system, but of course they together. So here is some outcomes from such a model. We can see 4 time snapshots here, and in every pair of figures, the left one is the cell layer, and the right one is the virus layer. And you can see at the beginning, there are a few red dots here and there in the green ocean. So greens are the uninfected cells, and the red dots are few infected cells. And of course, there's not much virus, so that's very black at the beginning. If you go to the second one, then you can see from these small dots, infection spread around in the neighborhood of those cells, and they started to produce virus. So you could see these red areas over there. And if we go forward in time, you can see how this infection spreads across the whole cell population, and eventually most of the cells will be infected or dead at the end. So with this model, we could reproduce some experiments from the literature. So here you can see 2 rows of figures. The top row is from an experiment, so it's from a paper that describes the experiment. Here these green spots denote, they mark the virus, and the bottom row is our results from our simulations. You can see we could get very similar outputs from computer simulations than the real experiment was. And we could also reproduce curves like this in the context of Paxlovid, when you use different concentrations of the drug and measure what fraction of the cells will remain unaffected during the infection. And on the right side, you can see experimental results, and the left side, what we get from the model simulations. And we published a few papers about this in these journals, but I don't want to talk about our results in this talk. It's more about an invitation for the future. So I've been thinking a little bit about the potentials of this in silico modeling in the context of virology, and there are a number of ideas where this modeling approach can be useful. So first, it has explanatory power. It can explain why these curves that we observe look exactly how they look like. You can also use in silico modeling to optimize your experimental design. Maybe you can a priori estimate what range or what concentration is suitable for your experiment, and you will never run an experiment when you add too few antivirals or too much antivirals, and the experiment becomes not interesting, or same with number of viruses and so on. So it can help optimizing the experimental design itself. That may lead to fewer failed experiments, and all of your experiments will be more meaningful. It can be also used for parameter inference, so we can estimate parameters which you cannot directly measure. It can be infection rate, or the viral diffusion rate, or lifespan of the infected cells that we have seen in the previous paper, or duration of the eclipse phase. So these kind of insights you can also get. You can also understand if you change something in experimental setup, why the outcome changed in the way it did. Then we have one ongoing project with my colleague at Virginia Tech, where we try to answer this question is, can you identify the mode of action of an antiviral from just looking at the viral trajectory? So if you see a different curve, can you tell from looking at this curve that this antiviral must have this kind of mode of action, so it prevents or blocks viral entry to the cell, or it blocks viral reproduction, viral production. So what is the mechanism of action here? And also, of course, you can run lots of virtual experiments, and in the virtual experiments, you can use variables which are unobservable in reality. For example, size, or how many cells you have in a given intracellular stage. You may never be able to measure it, but as part of the virtual experiment, you can have these variables. And also, you can explore sensitivity to different factors and identifiability. Can you identify which mechanism was important here? Which elements are the best predictors of the outcome of the experiment? And also to understand variability. So is cell heterogeneity important in the outcome or not? So with all this, of course, this in silico experiments, they will never replace real experiments. Of course, science must be based on hard empirical data in the first place, but they can amplify these experiments. So they can help in a number of ways to optimize these experiments and make them more efficient. So that's what we are trying to do with or putting together a small team for this year with Gabor and his colleagues from here and my modeling colleagues in Szeged, and try to run a pilot project where we can see how in silico modeling can work together with real laboratory experiments done here at the virology lab. So if you find anything of these potentials useful, or you have maybe other ideas how this can be useful, I'm very welcome to discuss with you later at this conference or any time in the future. So thank you very much for your attention.
Thank you for the presentation. I think I can say it for all here that if we hear fewer failed experiments, we are all ears. Please, sorry, it's a bit too big. Please welcome Professor Norbert Pardy. I would like to invite him from the University of Pennsylvania to give his presentation.
Thank you so much for the invitation. It's really my great pleasure to be in Page and tell you a little bit about our studies on mRNA vaccines. My time is very short, so this will be more of a teaser rather than a full presentation, but my lab is very collaborative. We have more than 100 collaborators worldwide, so if you believe that we can work together and come up with something cool in the future, feel free to reach out to me. This cartoon shows the most advanced mRNA-based vaccine platform. We call this nucleoside-modified and purified messenger RNA formulated into LNPs, lipid nanoparticles. This is the basis of the COVID-19 mRNA vaccines developed by Moderna and also Pfizer-BioNTech. And these are the vaccines that we also use, that basically we developed at the University of Pennsylvania more than 10 years ago, and now we can use this platform for many, many different purposes beyond vaccines. So the major research focus of my lab is infectious disease vaccines, but we also develop cancer vaccines using the platform, and again, you can use it for many, many applications, such as protein replacement therapy, but it's also gene editing, if you wish. So this is more of a platform. So the platform has two basic components. the protein encoding messenger RNA, and the second component is the delivery molecule called lipid nanoparticles. And the big invention, actually there are two big inventions in my opinion in this platform technology, and many little inventions. The two most important inventions is the introduction of modified nucleosides into this messenger RNA. So instead of uridine, we use, okay, good. So we use N1-methyl pseudouridine. This invention was basically introduced by Katalin Karikó and Drew Weissman from the University of Pennsylvania, and they received the Nobel Prize in 2023. And the other invention is the lipid nanoparticle. So many times people don't talk about it a lot, but without these special delivery molecules, the vaccines wouldn't work. This is important. So I was a bit surprised that those people who developed these lipid nanoparticles 20 years ago, they didn't receive Nobel Prize yet. So anyways, the point is that, and this is what I just want to tell you today, that you can optimize both the messenger RNA component and the lipid nanoparticle component for different purposes. Today, again, the major focus is vaccines. I won't have time to talk about the messenger RNA component, but again, you can introduce many different changes, many different little tricks. You can use different types of mRNA chemistry. Instead of N1-methyl pseudouridine, you can introduce other types of modified nucleosides. Or you can optimize the messenger RNA itself, the UTRs, the untranslated regions shown with red in this cartoon. You can also play around with the poly-A tail, and you will get a very different type of RNA with different immunostimulatory features. You can do the same thing with the lipid nanoparticle, and I will tell you very briefly about our relatively recent studies. Some of them are very recent, some of them were performed a couple of years ago. And again, I'm going to show you some data. So now there are thousands of papers showing that this mRNA platform works really well in preclinical models, but what is even more important, that it works very well in humans as well. But there are very few studies that really tell us about the mechanism of action of messenger RNA vaccines. And I think it's really worth knowing about it, because I think many of you received either the Pfizer or the Moderna vaccine. So it's just good to know how they work and how they can induce very potent immune responses. So I'm going to show you a couple of slides about one of our papers published in Immunity a couple of years ago. And after that, I will finish up with really just 3 or 4 slides about our very recent findings in this topic. So when I started my lab, now 6 or 7 years ago, I asked a very simple question. And the question is here: what provides the adjuvant activity for modified and purified mRNA LNP lipid nanoparticle vaccines? So again, I asked this question well before the pandemic, and I think no one really cared about it that time, but I was very curious how these types of vaccines can activate the immune system and induce very strong immune responses. Adjuvants are immune stimulatory molecules that help to induce a protective immune response. There is an adjuvant component in every single vaccine. In some vaccines, such as the virus-based vaccines, live or dead, the virus itself, the different components of the virus, provides adjuvant activity. In protein subunit vaccines, you will need to add some kind of an adjuvant. And again, also in mRNA vaccines, there is an adjuvant component, but again, until pretty recently, people didn't really know how these vaccines can induce immune responses. So since I mentioned to you that these vaccines have 2 basic components, the modified RNA and the lipid nanoparticle, the answer to this question can be the adjuvant activity stems from the mRNA, the lipid nanoparticle, or from both components. To address this, or answer this very simple question, we put together a very simple experiment. We immunized mice with recombinant protein that was an HA, hemagglutinin protein. Hemagglutinin is the major surface glycoprotein of influenza virus. And we just immunized mice with the hemagglutinin protein from the PR8 strain. This was just protein, no adjuvant. The second group received the protein with empty lipid nanoparticles. ELMP stands for empty lipid nanoparticles, so there was no mRNA cargo in this compound. And the third group received the HA protein mixed with the firefly luciferase encoding messenger RNA lipid nanoparticles. So this was basically luciferase, an irrelevant mRNA. We just wanted to see if mRNA has any immunostimulatory component. So these mice received the IM, intramuscular immunization, with 10 microgram protein, and 4 weeks and 8 weeks post-immunization, we measured HAI, hemagglutination inhibition titers. That tells you about the quality of antibody responses. And then it turned out that even the empty lipid nanoparticles shown with green could induce pretty nice antibody titers, showing that lipid nanoparticles have a strong adjuvant activity. This was really not known until recently. The reason why people started to use lipid nanoparticles for mRNA vaccines because they wanted to protect messenger RNA from degradation. So the lipids basically shield the mRNA, and they really help with delivery. They really facilitate cellular uptake. But again, until recently, we didn't really know that they have a very strong immune stimulatory activity. Later on, we repeated these studies, and we added another comparator shown with blue. It's called Adavox. Adavox is an adjuvant that is very similar to MF59. That is actually an adjuvant that is used in humans. So this is a relevant comparison. And it turned out that the lipid nanoparticles induced higher antibody titers compared to the, again, human adjuvant Adavox. So again, from this very simple experiment, we learned two very important things. One of them is that the lipid nanoparticles have adjuvant activity. The second thing that we learned is that we can use lipid nanoparticles adjuvants not only with messenger RNA, but even with proteins. We wanted to learn more about the mechanism of action, and again, I'm not going to be able to show you too many details, but you can check out these papers if you are interested. So first of all, the lipid nanoparticles that are being used for mRNA delivery are different from commercially available liposomes because they have a magic component. It's called the ionizable lipid. This ionizable lipid is very important for the safety of these lipid nanoparticles. You cannot use liposomes in humans because they would induce a very, very high level of inflammation, so people wouldn't tolerate it. But this ionizable lipid makes LNPs very safe. We just made empty lipid nanoparticles with ionizable lipid. This is the green, and the brown shows the empty lipid nanoparticles without the ionizable lipid. So they are relatively similar in size. And we repeated this study, this immunization study, using HA protein mixed with the empty lipid nanoparticles or empty lipid nanoparticles without the ionizable lipid. And as you can see, if you leave out the ionizable lipid component, there is no immune response. So basically, the ionizable lipid is critical for the adjuvant activity of these lipid nanoparticles. Learn a little bit more about the type of inflammation that is elicited by lipid nanoparticle adjuvants. We immunized mice with Adavox, empty lipid nanoparticles without the ionizable lipid, and luciferase mRNA LNPs. And groups of mice were sacrificed 4 hours and 24 hours post-injection. And we looked at the cytokines and chemokines, inflammatory molecules that are induced by these different formulations. And you can appreciate that there are many types, many different interleukins and other types of inflammatory molecules induced by these compounds. But only the empty lipid nanoparticle that contains the ionizable lipid and the luciferase mRNA LNPs induce this very strong immune activation shown. The deeper the blue, the higher the immune activation. And we were very surprised to see that there is one interleukin called interleukin 6 that is very potently induced by empty lipid nanoparticles or the luciferase mRNA LNPs. Interleukin 6 is a critical component of T follicular helper cell biology, and T follicular helper cells are critical for antibody responses. And as you know, for most viral vaccines, you primarily want to go for antibody responses. You heard these terms that antibodies, neutralizing antibodies, this is critical for most viruses, not for everything, not for all infectious diseases, and definitely not necessarily for cancer, but for viral vaccines, it's important. So we looked at the role of this LNP-induced IL-6 in a little bit more detail, and we did a very simple experiment. We immunized mice with hemagglutinin protein mixed with empty lipid nanoparticles, and then 10 days later, we looked at TFH, T follicular helper cell responses. But before we did this immunization, these mice received one group of mice received this anti-IL-6 antibody treatment, just to fully block IL-6 production. The control group received an isotype control antibody treatment. And then it turned out that in those mice that received the anti-IL-6 blockade, there is a blunted TFH cell response shown with the orange symbols. So again, lipid nanoparticle-induced IL-6 is critical for induction of strong T follicular helper cell responses in mice, and most probably in humans as well. So to sum up this part, we showed that the lipid nanoparticle component of mRNA vaccines is a strong follicular helper cell, B cell, and humoral immune response inducing adjuvant. LNPs can be used as standalone adjuvants for protein vaccines as well. Deionizable lipid is very important for the adjuvant activity, and LNP-induced IL-6 is critical for TFH cell response, for TFH cell responses. So in the last 2 or 3 minutes, let me tell you about our more recent studies and show how we proceeded with these studies. So in all the studies that I showed you so far, we used a single lipid nanoparticle. But you can make tens of thousands of different types of lipid nanoparticles with different chemistry. So this means that different LNPs have different adjuvant activity. And as I mentioned to you, for vaccines, it's critical to have this inflammatory activity because this is how mRNA vaccines can activate the immune system, if you will. But for different types of applications, for different types of vaccines, viral vaccines, bacterial vaccines, or even fungal vaccines in the future, or cancer vaccines, you want to induce a different type of adjuvant activity. And the big question that we asked a couple of years ago, can we really rationally design lipid nanoparticles for different purposes? Because that would be excellent and very, very useful in the future. So I already mentioned to you that these lipid nanoparticles are not homogeneous, so they have 4 components. Again, the big invention is the ionizable lipid, but they also have 3 other components, the phospholipid, cholesterol, and the polyethylene glycol PEG-related lipid. All of these components are important to form messenger RNA lipid nanoparticle complexes. And these are the typical molar ratios that I just indicated, 50% for ionizable lipid, 10% phospholipid, 38.5% cholesterol, and 1.5% PEG lipid. These are the molar ratios that are also used in the COVID-19 vaccines, both in the Moderna and the Pfizer vaccine. So what is really interesting that even, again, thousands of publications came out with different LNP optimizations, but I was very surprised to see that 3 years ago when we started this project, nobody really looked at the adjuvant activity in different LNPs with slightly different compositions. Moderna published a couple of papers and they've tested a bunch of ionizable lipids, but nobody really looked at the other components such as the phospholipid identity or the impact of PEG lipid ratios on the elicited immune responses, and this is what we did. This study was published in Nature Nanotechnology a couple of months ago. You can look at the details if you wish. I just want to show you a couple of, just very briefly, some data slides. So we tested 4 different LNP formulations in this study. This was the basic formulation. We just called it LNPQ because this contained the different components in the typical molar ratios, 1.5% PEG lipid, 50% ionizable lipid, 38.5 cholesterol, and 10% phospholipid. The phospholipid was DSPC. It's a phosphocholine containing formulation that is a neutrally charged phospholipid. I don't have time to tell much more about the background, but we just very briefly tell you about the 3 other formulations. So in some earlier pilot studies, we found that LNPH that has pretty much the same compounds as LNPQ. The only exception is that it's a very little thing. Instead of 1.5% PEG lipid, we only have 0.5% PEG lipid in LNPH. This is only major difference, but we found that if you decrease the PEG content, you can induce better antibody responses. So that's why we introduced LNPH into this study. And we also wanted to test 2 other types of phospholipids in these formulations. Again, instead of DSPC, that is a neutrally charged phospholipid, we introduced DOPG into LNPY and DOPS into LNPW. DOPG is a phosphatidylglycerol type phospholipid, and DOPS is a phosphatidylserine type phospholipid. Both of them are negatively charged. So we immunized mice with these 4 different formulations, Q, H, Y, W, and we looked at antibody responses in mice. And we were surprised to see that we did a good amount of difference in binding antibody titers in these mice. And what was particularly surprising to us that there was a huge difference between Q and H. So again, the tiny difference, just 0.5% PEG instead of 1.5. And at this time point, I think it's a 2-fold difference in antibody titers, but at later time points, this is even higher. So this means that if you modify the compounds or percentages, or even the type of compounds in lipid nanoparticles, you can induce different immune responses. These are just the neutralizing antibodies showing the exact same trend. So again, in these studies, it seems that LNPH with the lower PEG content and DSPC phospholipid, it induces the highest antibody responses. And later on, we showed that LNPH induces also the highest memory responses, MBC, memory B cell, and LLPC, long-lived plasma cell. Plasma cells produce antibodies, long-lived plasma cell responses. And I think this is the last data slide I'm going to show you. So as many of you, or maybe all of you know, that the immune system has 2 basic, the adaptive immune system has 2 basic arms, the humoral immune response and the cellular immune response. So I told you about the humoral immune response. We found that LNPH induced the highest antibody response, but the cellular immune responses are also critical for many infectious disease vaccines, and they are extremely important for cancer vaccines because CD8 T cells can clear tumor cells. So we repeated our studies with these 4 formulations in mice, and this time we looked at CD8 T cell responses 10 days after a single immunization of these mice with a spike mRNA LNP vaccine. So basically this was a COVID vaccine, similar to the previous studies. And we were very surprised to see that in this study, another LNPW that induced the highest LNPW induced the highest CD8 responses. LNPW contains a negatively charged phospholipid, phosphatidylserine. And I didn't want to believe this, so I asked my postdoc to repeat it a couple of more times, but in 3 or 4 independent studies with independent batches of mRNA vaccines, he always received the exact same data. So I'm going to stop here and just want to, again, very quickly summarize this second part of my talk. The point is that we clearly showed, and now others confirmed our findings, you can play around with the composition of lipid nanoparticles, and you can induce a different type of immune response. Obviously, we didn't have a time to write down every single rules that this is how you need to modify, but we clearly showed that different LNPs have different adjuvant activity. So I do believe that in the next couple of years, we can optimize lipid nanoparticles for different applications to induce the right type of immune response and come up with excellent mRNA vaccines for different applications. And this is the take-home of my talk. So with that, I'm going to stop here. Thank you for your attention, and I'm happy to answer any questions now or later. Thanks. Any questions? Okay, no problem. We can talk. Yes. My personal opinion is that PEG is not a huge problem. There is a lot of there are many publications about it that anti-PEG antibodies can cause issues. I think in the context of mRNA vaccines, it's not a gigantic problem. In some cases, it did cause prophylactic shock, but the numbers, if you look at the numbers, when we talk about the safety profile of the mRNA vaccines, it's 1 out of 30,000 people. So again, it happens, and I don't say that we shouldn't optimize these vaccines, but I don't think that without the PEG, these vaccines would be stable enough. It's very, very important to stabilize the lipid nanoparticle, and again, if you look at the safety data with these vaccines, and that's what many times journalists fail to mention, that this prophylactic shock, it's relatively rare. Again, 1 out of 10,000, 1 out of 30,000 people, and it can be revealed. In my view is that it's not a gigantic problem. People try to come up with LNPs that don't use PEG lipid. They try to find other compounds, but to my best knowledge, those are not as potent as the current generation of mRNA vaccines. No problem. Thank you.
Thank you for the presentation. I would like to invite Gergely Babinski from the Hungarian Defense Forces Medical Center to deliver his presentation.
So, dear colleagues, thank you for the invitation. I'm very glad to join you to celebrate the first 5 years of the National Laboratory. So let me start with a little introduction through the biodefense capabilities of the Hungarian Defense Forces. I'm from the Biodefense or Biological Defense Division of the Epidemiological and Scientific Research Institute. This slide can be much more complicated than this, but luckily we are directly subordinated to the Chief of General Staff of the Hungarian Defense Forces through the Commander and the Surgeon General of the HDF Medical Center. And again, we are working under the umbrella of the Epidemiological and Scientific Research Institute. And our main tasks or daily tasks relies on 3 main pillars, which you can see on the slide. One is the CBRM Biolab with own biological sampling team. So this is the core of our laboratory. We can provide CBRN, so chemical, biological, radiological, and nuclear. So we are the B, the bio side of the CBRN part, and we can perform biological examinations aiming potential biological warfare agents like bacteria, viruses, and toxins. We also have a so-called preventive medicine team, which is basically a laboratory on a truck, and it's a highly mobile asset, and we can give any kind of epidemiological and for self-protection help, for instance, in case of disaster relief missions and so on. And we also sorry, have a deployable clinical diagnostic laboratory. Here you can see the quite diverse tasks of our institute. So as I mentioned earlier, we have CBRN biological laboratory and biological sampling team. We also have a preventive medicine team, but we can perform medical intelligence tasks as well. We have other assets like mobile vaccination units. Our method development is continuous to be as effective as we can. And as you can see, within the Institute, there are routine laboratory tasks like drug and alcohol screening and various epidemiological and public health laboratory tasks like water quality test, the classical microbiological work, and occupational health and radiological tasks as well. We also have applied research projects connected to these laboratories, and we have other assets like scientific laboratory and so on, scientific library and so on and so forth. So yeah, it's quite diverse. I think basically this slide is the same as the previous, so we can skip it. And in the last few years, we just shifted towards this kind of public health issues in the civilian domain. And why is that? This is because of the NATO CBRN defense concept, which is slightly changed in the last few years, because before that, we needed to focus and concentrate the potential biological warfare agents itself, and nothing else. And after that, the decision makers realized that it's much more than that within CBRN defense concept. As you can see, now they include a broader spectrum of issues, including naturally occurring, accidental, and deliberately caused biological hazard. And that's why we need to work together with the civil side. And I think this is the key to be as effective as it can be. So that's why we appreciate a lot the cooperation with the National Laboratory of Virology for over a decade. Allow me to highlight a few examples from this cooperation. In 2019, sharing nucleic acid samples from the first COVID-19 cases confirmed in our laboratory, we contributed to the first phylogenetic and the phylogeographic analysis of the virus in the region. In 2018, we provided field diagnostic background facilitating the later analysis of the Yovio virus in Hungary. Through civil-military cooperation, or regular joint trainings could further enhance interoperability and improve national biodefense capabilities. As participants of a forming European Defence Agency project, which you can see here, we are working together in trapping and identifying of the relevant mosquito species. And as for the future, a promising common tool for collecting airborne viruses could be either the Impinger technique or the Coriolis micro tool, which operates on the principle of cyclone technology, which our colleagues have already successfully used in various tasks. So that's concluded my presentation. Thank you for your attention.
Any questions?
Maybe later. Thank you.
Thank you very much. Last but not least, please join me in welcoming Tamás Csánk from the University of Veterinary Medicine and Pharmacy in Košice. Kos. Please.
Thank you for the word, Daniel. First of all, let me thank to the organizing committee for this invitation, and I'm really honored to be here. And now in the next few 10-15 minutes, I would like to inform you about our results. I can say partial results about the molecular screening of West Nile and Usutu viruses in Culex mosquitoes in Slovakia. So West Nile and Usutu viruses, as we know, both viruses represent a zoonotic threat across Europe. And being arboviruses, their transmission cycle involves birds as reservoirs and mosquitoes as vectors which transmit the virus in the population of reservoirs. Occasionally, the virus can be transmitted to dead end or accidental hosts from which usually the virus is not transmitted. Year 2024 was significant from the point of view of West Nile virus activity in the human and animal population. And the figure shows us the weekly breakdown of number of cases during the period, during the transmission time, which spans from July to mid-October. Yes. 2025 was also significant with lower numbers. Now, we recorded that year, 2024, 9 cases of West Nile virus infection, which is, of course, in comparison with Hungary, more lower. Out of these, 6 were locally acquired. Oh, sorry, there is a mistake. So 6 were acquired, and for the first time, locally acquired Usutu virus infections were reported in humans. Slovakia lacks a national program of vector-based arbovirus surveillance, and as a part of an international project, we aimed to screen these viruses in mosquitoes which were captured during these years. So we performed continuous trapping from June to mid-October, or beginning to October. And as we can see, here quite big differences in the numbers of mosquitoes between the 2 years were significant. So 34,000 Culex mosquitoes versus 9,000 in 2025. According to Slovak Hydrometeorological Institute, the 2024 year was remarkable from this point of view because it was one of the most warmest year, and with above average precipitation. So this very likely aided the development of mosquitoes in that year. Now, the arrows and the circles points out the localities with the highest abundance of mosquitoes. And as you can see that these are located in the southern parts of the Danubian, the Danubian lowlands and the eastern Slovakian lowlands. So roughly we processed 47,000 mosquitoes in 1,200 pools, which were screened for virus RNA, either for West Nile or for Usutu virus. And out of these, West Nile virus RNA was detected in 7 pools from 4 localities, which, as you can see in the map, are these double-colored circles, and can see that they copy the Slovak-Hungarian border. So on the southern part, West Nile virus occurred. On the other hand, Usutu virus RNA was detected in 64 pools in total during both years. And except 2 countries or localities, here in the north and northeast, the virus RNA was present in each locality. However, simultaneous study carried out in the same way by the colleagues from Slovak Academy of Sciences in collaboration with regional public health institutes, they found out that the virus also, Usutu virus, also was present in these localities in the north and northeastern parts of Slovakia. Oh, sorry, this is not the updated version of my presentation, so my apologies for this. What I want to point out further from this presentation is that West Nile virus was detected only in year 2024. All the pools were negative from 25, but Usutu virus was present in both years, and there were 9 localities in which the virus circulated during both years. Now, these positive pools are not from the same date, but from different dates, so this suggests that the virus endemically circulates in those areas. Characteristic for arboviruses in general is their diversity, the genetic and also serological diversity. So West Nile virus can be divided, the lineage 2, because lineage 2 is characteristic for Europe, can be divided into 4 groups. And out of these, the F group is further divided to 2 subgroups. Each of these strains or subgroups encompass strains with characteristic or certain geographical distribution. Now, we work on West Nile virus roughly 10 years, and our first isolates are from years 2013, 2014. And these are avian isolates, and these belonged to the E group, which are characteristic for strains which are closer to the Western strains, Western European strains, and Central European strains. Now, 2018 was also significant from the point of view of epidemiology of West Nile virus, with the highest case number either in humans or in animals. And from 2018, we have also avian strains. Not because we didn't detect in mosquitoes, but we just start to work with the mosquitoes in 2024. So after the 2018, we detected mostly only strains, West Nile strains, which are genetically closer to the southeastern Europe or Western Balkanian strains. There was only one strain which belonged to the D1 group. Excuse me, this one here, which belonged to the D1 group, which was more closer to the Greek cluster. The same is true for Usutu virus, also genetically diverse, and it shows some mosaicism across Europe from the point of view of genetic lineages. Now, in the surrounding countries of Slovakia, there are mainly European lineages. However, in some countries like Austria, Czech Republic, and also recently published paper in Poland, African lineages also occurred. However, these were mostly detected in avian carcasses. Now, in our study from 2025, we detected European lineages, namely lineages from the group Europe 2, and one strain which belonged to the group number 1. And this confirms the previous study, in which mostly European strains, strains from lineage, Europe 2 lineage were detected. Now, for conclusion, based on the data from the Hydrometeorological Institute, we can say, and that's generally known, that this high abundance of mosquitoes is aided or is very likely due to these weather abnormalities, as 24 was a very high year, and with also precipitation abnormalities. For our surprise, Usutu virus was really geographically widespread across Slovakia, almost in each locality. And West Nile virus was located more on the, not more, but only on the south, on the south part. But also surprising for us were the high minimal infectious rate in Usutu virus, which despite the low number of mosquitoes during 25, still remained almost the same as in the year when the abundance of mosquitoes was really high. This mosaic occurrence of different virus lineages is characteristic for both of these viruses and in general for viruses. And it is, I can say, heavily influenced with two properties. First one is the local evolution of viruses in a certain locality or in a country. And the second, in case of these viruses, is the long-distance introduction by migratory birds. And what I want to say here with this is that I think that for a more precise or realistic picture of this geographical distribution of different lineages, we should not focus only on one part of the transmission cycle, but take it as it is, as a whole. Focus on the vectors, focus on the reservoirs, either alive or dead carcasses, I mean avian carcasses, and also for the accidental host, either humans or horses or other mammalian species. So, and at the end, I would like to thank to my colleagues from the University of Veterinary Medicine, also from Slovak Health University, also from NVL, and also for colleagues from Veterinary and Food Institute in Košice for their meticulous work, because really to sort out from those nets, which was in these localities, it was quite a laborious work. So thank you for your attention.
Thank you very much. Any questions? Thank you. Then I believe we have reached the end of this session. The weather in Pécs is partly cloudy with a low of 9 degrees outside. After this, there will be a coffee break for 30 minutes. Please feel free to eat, drink to your heart content. After that, we will begin the next session at 15:30, so 3:30 as scheduled. Thank you very much.
Start the last section, okay? Hú, de hangos vagyok. Okay, so I welcome everybody again. I will open the last section of this day, and I call myself because I will be the first presentation. Yes, with the first presentation. Okay, so the antiviral drug discovery often begins with very fundamental researches, but only a fraction of these discoveries reach very patient-able innovations. So with my presentation, I will take you through a little journey from the basics to the innovation property, focusing on antiviral advance in glycopeptide antibiotic derivatives. In my previous presentation, I mentioned and I showed that we start antiviral compound testing activities with these antibiotics and derivatives. We work with the collaboration of University of Debrecen, and we have many teicoplanin, vancomycin, ristacetin derivatives and probe them at first against that West Nile virus, and we find several promising candidates. During the COVID-19, we also worked these compounds, and they also show very strong antiviral activation. And after we took one interesting question, what happened when we probed them against different viruses? And we take that, we included into the experiments flaviviruses, alphaviruses, Zika virus, chikungunya simpis, and onyonyong virus. And after we optimized every stages of our workflow, so the virus stock preparing, the determination, the infectant doses, determining the cytotoxicity of the antiviral agents, we use viability assays, immunofluorescence stainings, and molecular biology-based readouts, and evaluate their inhibition activation. At first, we worked with the Zika virus, and we found that several teicoplanin aglycon derivatives were very promising and exhibit inhibition activity at very low concentration. When we worked with the chikungunya virus, we found the near same findings. In the case of the 6th and the 7th compounds, they also show very low effective infective concentration values. During our work, we used different assays which complemented each other and allowing for us to confirm the inhibition activation of these compounds. And after that, we work with the compound 7, and with the immunofluorescence assay, we can determine also again the antiviral inhibition activity of these compounds, which was a teicoplanin amide perfluorohexyl derivative. And after we determined that this derivative strongly inhibits all 4 viruses in vivo cell line. And also we tested in a human cell line, and we found a similar effective concentration values as well in the viral cell. And there will be 2 interesting questions. What could be the mechanisms of this action, and where do it or interact in the pathogenesis of the virus? For this, we used the time of addition assay when we treated with the antiviral pre, co, and during the post-infections in the timeline, and the results indicated a significant differences in viral RNA level compared to a non-treated group, and only on the case of the co-treatment leading us to conclude the compound experts its effect during the early stages in infection. I'm sorry, I forget the graph. After we get help to explore further mechanisms with the bioinformatic analysis, after the FT map scanning on the viral A protein, we found 3 sites where the compound is able to bind, and all of them take part to the inhibition between the lipid membrane of the endosome and the viral envelope. So if I summarize everything, all of experiments that precede analysis and the microscopic experiments also support the model. Therefore, we propose that the site 1 is the main binding site of the compound with the inhibition of the E protein, dimer-trimer conversation at the basis of the detecting in vitro antiviral activity. After we summarize everything, and our results demonstrate that the investigated semi-synthetic aglycone triazole perfluorohexate derivative, the LA116, exhibits a broad spectrum antiviral activity, and these findings highlighted the antiviral, new novel antiviral ingredient. So we were preparing a patient application, and it's covered the antiviral uses. For this work, I say many thanks to Ilona Beretsky and Aniko Borbás, and the 2 excellent colleagues, Tina and Zoli. So thank you so much, and for you, thank you for your attention. Okay, I will call the next presenter, Roland Hetényi from the Rolling Biotechnology company.
Thank you. Hello, good afternoon to you all. Well, it's quite a pleasure to be here. So it's an anniversary of the Virology Department, the National Lab of Virology, indeed, and my story started out with the National Lab, indeed. So this is the art of the kill. Back in the old days, in 2020, with Daniel, my best friend in middle school, we started out with an idea. We couldn't really start to work with the virus. So we had to somehow get to the virus. And the idea was to inactivate it. So this is Daniel, who I started out with. And at the moment, we are so highly esteemed that we have partners like the Hungarian Defense Forces or the European Defense Fund project with Professor Kurt Zatluka, who's sitting among this very rows. And we have a nice, indeed, team to balance our strategy on. And as my colleague Daniel earlier presented, we are a regional racer. So the only BSL-4 in the Central European area is located here in the building, and that's the one that we are very keenly building upon. So the problem we started out with in 2020, 5 years or almost 6 years ago now, is that these BSL-4s and BSL-3 labs are very scarce in the world and very hard to get to the virus. And we wanted the virus so bad. SARS-CoV-2 was a BSL-3 virus, and it was very hard to work with, but we got great ideas on how to... so BSL-3s contain SARS-CoV-2, but we at PH have a BSL-4 only. And Professor that year told me that you want the virus out of the lab. You're crazy, right? That's the one that goes into pandemic. So we had to, of course, we don't want the live virus, we want to inactivate it. And this is what happens. So we came up with the idea of UV inactivation because we wanted the virus in its early shape with no modification at all, but completely safe. And this is what we did. We managed to come up with an idea and a device, because you just can't buy a UV inactivation device for all BSL-4 labs in the shop. And we built it. And we also made some patenting application using that device. And thanks to our friends at the military, And at VNR2, they measured a few parameters with the inactivated virus. So from the active virus, we used our device and technology, the way we built, UV-C inactivation. And what we saw were quite astonishing. So with the red bars, you could see the active virus, and with the blue bars, you could see the UV inactivated virus at the same concentration, in the same media whatsoever. So this is an ELISA assay showing us that there was no change in the RBD protein segments after inactivation. And this is complete inactivation, which was great, honestly. We repeated the experiments, we repeated with other viruses, and it always showed up the same. No change at the proteomic level. Well, we could do our projects. And another thing, which was quite nice, that we wanted to test whether the PCR was intact, whether the nucleic acid, what we use in the everyday's life, could be tested as if the active virus was there. And we found out that yes, we can. So the active virus is once again red, and after inactivation, complete inactivation occurs after 30 seconds, usually, using this setup. And in multiple dilutions, we could see, that's from a pipetting error, but we could see that there was no change, and after statistical analysis, we also showed that there was no change in the PCR readings for the completely inactivated virus. And this is the slide we won an EDF project with. So the military was so keen on this slide that we could enter these kind of markets. And I made some assumptions on how to compare visually the chemical and heat inactivation with the UV inactivation. And what we could see is that with conventional chemical and heat inactivation, you will see damaged proteins and also PCR readings. And of course, they are known and in use and cheap, so most people know. But on the other hand, UV inactivated viruses and the UV inactivation technology will give you 100% of the protein of interest and 100% of the PCR readings. Great for diagnostics. And it is very cost efficient and in line because we've just upgraded our system to an industrial level, which is great. Yeah, so our main key of interest now is of military diagnostics and European Defence Fund projects, but we also tend to have other uses, and these other uses include vaccine candidates, because a nice first generation inactivated virus in itself is a nice vaccine candidate. These are the already inactivated viruses so far, quite a dull list. And I've got some slides which are very technical, and Daniel would explain if he was here. But anyways, I wanted to show you some animal experiments, because I had the idea, okay, if this is such a good thing that the protein is intact and the nucleic acid doesn't really change in the PCR readings, that can it work as a vaccine back in 2021? And I found out that yes, there was no body weight change in Syrian hamster model, it is safe. There was known as a pharyngeal ddPCR reading, meaning there is no replication in the throat of the animals, meaning safe. And we came up with a ELISA measuring the Th1 response, an IGRA, as you would say, interferon gamma release assay, from the cells. And the reading for the first time, through a nasal input to the Syrian hamsters, we could see that there was a high signal with the active SARS-CoV-2, but there was some measurable and intently high signal with the UV inactivated part too, and that was a control. So we could measure that indeed it works, so it could work as a vaccine. So we were quite happy. And we also made some virus purification, we patented it, we gave it to the university, it worked, it's amazing, we could use the same in military applications. It is the methodology that is including here and will be used, otherwise we have a business model now that we operate in. We contact large projects like EDF for Hungarian grants, and we also want to disperse and produce inactivated viruses to democratize virology, so that other people like us before in 2020 could not access the virus that is of interest. Other people could by using UV inactivated virus. In the literature, other people too find it very interesting to do UV inactivated vaccines. They are on the rise, but the industrial scale is of very high difficulty. And for now, we are the only ones in the world who can do that. This is the art of the kill. Thank you. question, please. So yeah. So the question was about sequencing and whether sequencing could be done. And the answer is yes, we've done some nanopore sequencing, meaning that the consensus sequence remained the same, but we could alter differences in the amplicon region. So amplicon readings became altered, but the consensus sequence is the same. So if you are using it for field diagnostics, then a UV inactivated sample could be used for sequencing. If there is very minimal sample quantity, then other measures need to be taken care of. But yes, it's a viable option, but it can be done, yes. Any other questions, please? Lovely. Well, thank you.
Presentation. Működik, nem? Sorry. So the next presenter, Gábor János, from the Captain Medical Company.
Thank you very much, Annette, for the introduction. It's pretty hard to talk after Dr. Hetényi, who is pretty good in science and business as well, but I try to do my best. And we have been asked to talk about the business perspectives of the basic research, and my personal opinion that it's not possible to make direct borders between fundamental research and business, because what we do in the medtech or in the deep tech, these technologies really require us to still stand on the fundamental research part, but we should somehow change our mind and try and find out how we can use what we have developed in the laboratory, how we can use in the real life, how we can make a product, and we should not be so shy, how we can make money on that one. And if I'm right, if you have seen the memorial board in the entrance hall about Professor Jakob, he mentioned that the results should come out from the laboratory, and I really agree with him that the results should come out of the laboratory. And this is what I would like to try in my presentation, the balancing between business and science. So every startup pitch starts with, what's the problem? It's very easy to remember back to the 2021 when we have the COVID pandemic, and we have seen terrible numbers, so many cases, identified cases, and so many deaths due to COVID, which is terrible. So I should not create here the hell. The hell is here. We have seen again, and we have experienced. We can remember very clearly that at that time we had no good methods how to deal with the new pandemic. And at that time, our company was focused on a very different field. That was the oncology field. We have developed the technology to remove the circulating tumor cells from the bloodstream. And for that, we had an extracorporeal device. And due to the pandemic, we were forced to try to figure out how we can modify our cartridge to remove the viral particles from the bloodstream. With this very simple approach, we get to the idea that we have to try to develop a cartridge which will remove the virus particles from the bloodstream. It is so simple. But again, the original ideas came from the CTC field, where we wanted to remove the CTCs from the bloodstream. It was an immune affinity-based technology, and originally we wanted to modify hemodialysis semi-permeable membranes to be able to capture the CTCs. Why we wanted to modify the cartridges instead of developing something new? Because all of the existing pipelines were there, so it's way much easier to implement something new into the pipeline when you have the supply chain, when you have the machine there already, when the operators are already trained to use the device. So we wanted just to replace the conventional hemoperfusion cartridge into one which can act like an artificial organ, which is removing the CTCs. You can see in the slides what was the shape of the removal process. And that was very successful. We have published the scientific background, the technical background of the product, as well as we have patented that technology, which is important from business point of view, because do not forget what I would like to emphasize right now, how we can do the transition from the laboratory until a product. Of course, we have identified how we can capture the CTCs. What you can see in the slides on the left, these illuminating small points are the green fluorescing CTCs that we have captured with the modified hemoperfusion system. And in the SEM images, you could see that the original structure of the semi-permeable membrane was intact, meaning that we can do in parallel two effects. One is capturing the CTCs, and the second one is that it's remaining the original function, so it's still semi-permeable, so it still can remove those small substances from the bloodstream. And that was the background how we started to think about this virus capture product. Again, we were forced. It was a pretty good motivation that we had this COVID pandemics. I guess it's very much more familiar for you, this figure. Actually, we have seen this already in the morning. This is a computer simulation about how the spike proteins looks like. And there is one point what I would like to emphasize here, that those glycan or sugar structures on the surface of the spike protein are coming from computer simulations or in synthetic way. According to my best knowledge, there is no current technology which can investigate the actual end glycan profile of a certain virus. And here I have seen a pretty good opportunity to putting together the pieces of the puzzle, what I'm talking about. We have to have the technology from Dr. Hetényi, which is producing us the intact, the inactivated viruses. We have back in West Point a laboratory which can separate and identify the end glycan structures of from various sources. So if we put together this, then we can result in a technology which is going to be, in my understanding or in my opinion, the first technology ever which can be used to in situ identify the end glycosylation of a certain virus. And if we develop this technology, then we could be prepared for the next pandemic. And I'm sure there will be a next one. So we should be prepared for that one. So I would like to use this short slide story to emphasize how we should think about the basic science and how we can address actual needs. That's my basic approach for doing that one. And 2 years ago, I was talking about how we have developed the virus capture technology. At that time, I have used that slides because back in 23, we had no patent protection on that one, but since that time, I'm very proud to announce that our patent application has been accepted very recently, so I can show you the next slide, which is a more clear view of what we did. Actually, it's a wet chemistry approach, how we are immobilizing the antibodies on the surface of the solid support. It's a wet chemistry. Of course, the devil is in the details, so I would not share you all of the reaction conditions and reagents, but the take-home message here that we developed a wet lab chemistry, wet lab process, how we can do the effective immobilization, how we can put capturing agents like nanobodies or antibodies on the surface of a solid support, which can be used to capture, based on immune affinity approach, how we can capture the viruses from the bloodstream. Here you could see the modified shame. It's very similar what I have showed you before for the CTC. Again, we have the solid support, the small beads, and on the surface of the beads, we have immobilized the binding agents, again, which could be nanobody or antibody, and it is just like key and the lock, we capturing the viruses and nothing else removed from the bloodstream, so then the clean blood can be given back to the patients. This is a very short slide about how we are modified not how modified, but how we have produced the antibodies or the nanobodies in our case for capturing the SARS-CoV-2. And yeah, that's the part where I have to emphasize how we have collaborated at that time with the National Virus Laboratory, especially Anat was the one who carried out these experiments, if I remember well. So I have to say thank you again. And with these measurements, we have proved that the new nanobodies that we have developed are effectively able to neutralize the SARS-CoV-2. And once we had the new nanobody, which is able to capture the viruses, and if you remember from the previous approach, we already had the device itself, so we were very easily able to put together these two pieces, and we have built up a virus trap. Of course, at the beginning, we did prototype miniaturized size. It's like a HPLC column. I'm not sure, do you? Okay, the HPLC column in the picture, you would see. And we have used that laboratory setup to test whether our approach will work or not. And these experiments were carried out in the BSL-4 facility up there. And just very shortly about the results, because again, I have been asked to do the highlighting the business part of this approach as well. So we were able to capture specifically so many virus particles, and if you consider the figures there, if we build a real size hemoperfusion device to remove the virus particles, we had the ability to remove more than 15 million genetic copies, which in certain cases allow us to get a therapeutic effect on the patients. Here we have proved how we have captured the virus particles, so you could see in different resolutions of the SAM images that we proved that we have captured the virus particles. There's another one. This is my personal favorite, albeit there is this black hole in the middle, but we were able to do this very nice picture about the captured SARS-CoV-2, and if you investigate it carefully, you will be able to see the S proteins as well. And I'm not sure the projector allows that, but believe me, definitely you can see in my slides. And again, as it's a brand new technology, and we were able to think back at that back time how that we would like to transfer the basic research towards the product. So we have submitted it for a patent application, and we are very proud of that one, that we had a valid patent already in the United States and in the European Union. And of course, we did publish the scientific and technical background of the project as well. And what is more important, that I do see a potential how we can further develop the technology, because again, definitely new pandemic will come up later or soon. So with that slides, I do not want to be too, to say, pessimistic. I would be very optimistic with the next one, because I have so many colleagues and cooperating partners. So I do believe that we could be well prepared for the next pandemic with this technology. And yeah, we should be very optimistic on that one. Thank you very much for your attention.
Do you have any question from Gabor? No. Okay, thanks. So the next presenter is István Németh from the Fluart Innovation Company.
First of all, thank you very much for the invitation. Today I will talk about how hard it is to develop something therapeutic that has bacteriophages, or the API is bacteriophage. First of all, who we are. Fluart was founded many years ago. We are the only company that produces flu vaccine in Hungary. Our vaccine is available for everyone. And we are a CDMO as well. And the most important thing for me is that we are developing products or pharmaceutical products that based on bacteriophages for the food industry, for veterinary use, and for human applications as well. Why do we need bacteriophages? The main reason is the antimicrobial resistance. Actually, the most important part is this one. I don't know. No, it's not visible. Just a second. Yes. This part, because it shows that in 2050 there will be 10 million deaths caused by the resistance bacteria, and the cost of the antimicrobial resistance will be billions of dollars. So that's why we need to do something with the resistant bacteria, and bacteriophages are really good tools, maybe, for these. So bacteriophages are basically viruses. They can have RNA or DNA as a genome. Mostly double-stranded DNA is the genome of the bacteriophages. And the most important thing about bacteriophages are their life cycle. So they can have a lytic cycle. Virus goes in, kills the cell, repeats itself, and goes out. But some bacteriophages can be in a lysogenic cycle when they enter the cell and integrate their genome in the bacterial genome and replicates with the bacteria. And for even years, they can replicate with the bacteria, and up in a certain stress, they cut out their DNA from the genome and become lytic or lytic-like, but they can integrate themselves again. And some rare cases, there are the chronic cycle where the production of viruses do not kill the cell. Actually, the history of bacteriophage therapy is quite long. As you can see in yellow, the usage of bacteriophages for therapeutical purposes, and with blue, the application of antibiotics. So in many countries, after the penicillin boom, the use of antibiotics goes up and the use of bacteriophages goes down. But for certain countries like Russia, Georgia, they kept bacteriophage therapy, and they developed it for more than 100 years, so they are ahead of us. And in the 90s or in 2000, the bacteriophage therapy became more important again because there were many resistant bacteria. And in the few years ago, there were some cases where people could be saved by bacteriophages. For example, in England, there was a patient with cystic fibrosis. None of the treatments were successful, but a bacteriophage cocktail of 3 bacteriophages could save her life. So, what are the current regulations in the EU? Actually, no regulation. In 2018, the Belgian monograph came out, which was the first paper or regulation-like something about bacteriophages for magistral preparations. Later that, in 2023, a concept paper came out, kind of guideline. It was published in 2024, or the debate ended in 2024, and after that, in the beginning of 2025, a draft guideline came out, but it's still a guideline. As you can see, in one page, you can see should 18 times. So it's not a regulation, it's something that you should do or shouldn't. So that's the problem with the regulation. And there are some other problems with bacteriophages in terms of using them as API. Narrow host range, even, for example, Salmonella infantis, Enteritidis, Typhimurium has different phages and no cross-reaction between them. Or for E. coli, each serotype has different phages. So somehow we have to broaden their host range. Resistance emerges quickly because bacteria, it's quite a coevolution between the bacteria and the phages. Receptor changes, CRISPR system, abortive infection means that the virus produces itself but cannot assemble, for example. We see in many cases that in a plate it works perfectly, but when we want to use an in vivo, it's not working. There are many factors that could make difficult for the phages to work in a system. Most importantly, we need lytic phages because all of these suggestions say that only clearly lytic phages can be used because they can horizontally transfer resistance genes if they are not lytic. For each phage, we need whole genome sequencing, and we need a lot of characterization for their function. That makes it difficult. To make the range broader, we can and we have to use cocktails. But in cocktails, phages can be antagonize each other, and it makes more difficult to use them like this. Dynamic cocktails could be useful, but like other drugs, other medicinal products, it's difficult for the regulatory point of view that if a new bacteria emerges, I can change one phage to another without clinical trials and so on. The pharmacokinetics and pharmacodynamics are completely different, since phages replicating inside the body and when they kill the bacteria, they are removed from the body by the immune system, but classical measurement of pharmacokinetics and dynamics is impossible. There are different parts of the body where phages could be killed easily, like biofilms, low pH of the digestive system, and many enzymes that can degrade phages, since they are protein enveloped DNA. The other issue is GMP manufacturing, because we have to define everything, define the phages, define the media, define the cells, and actually in the nature there are no bacteria that has no prophages, and prophages can be released into the culture media with the phages of ours, and it could be a impurity in the end product. The robustness of the process is difficult to maintain, and the characterization of the API, let's call it API, it's very difficult sometimes. We have to develop new methods for new phages as well. The formulation itself is quite difficult. For example, for oral administration, we have to protect them from the pH. For other type of administration, we have to protect them from other effects. The question is that we use liquid phase or lyophilized and reconstituted phages. And the other problem is that they attach every surface except glass. So the administration devices can be a source of phage number decrease. The other problem is the regulatory part, because in the pharma industry, standardized products, standardized production is the desired way of producing such medicine, but phages cannot be standardized. Bacteria changes, we have to change the composition, we have to change the phages. In some countries there are exceptions, something like magistral products in Hungary. For example, the pharmacists make some product for you in the pharmacy, and it could be used at home. The logistical challenge is also a problem, because phages need to be cooled. And the batches could be different, so that could be a problem. In many countries there are exemptions, local test, some ethical committee stuff, and for example, in the US, phage therapy could be used if the patient agrees. But in the EU and in Hungary, if the safety is not proven, the product cannot be tested in humans, even if they will die because of the lack of the treatment. And that's why there are not much clinical trials ongoing or finished. As you can see, only 4 trials in phase 1, 2, and only 1 in phase 2. And this phase 2 test is the famous Georgian Institute, the institute who develops phages for 100 years now, and their results are not proven to be effective, because as you can see, the indication is too broad. There's ampule of 5 milliliter phage cocktail, you can drink it and you can use it for 100 types of diseases, and this is too broad, and they could not prove that it's useful. And the ethical dilemma is, as I mentioned before, in the EU, we cannot test it unless it's proved to be harmless. Compassionate use, it's another question, but the financial point of view, the manufacturing cost is really high, and the other issue is that if I isolate phage from the nature, I cannot patent it. So it's not my phage. Anyone else could use if they want to. So we are quite far from developing phage therapy products for human, but we are working on it. So thank you for your kind attention.
Okay. The profile.
With my colleague Tamás, we are going to present you a company called Profil, which is located at Mohács, like 40 minutes drive from here. This is a family-owned company. We focus on industrial service business. So for this reason, we will be a little bit more marketing-oriented and less scientific. We have to preserve the intellectual property rights of our customers. However, Tamás will give you a little bit of background what we do in a more scientific way. So the company was founded 35 years ago by a couple, János and Benyeda and his wife, starting to focus first on veterinary clinical studies. I will tell you our services and activities later, so I will not go into details now. When János passed away, then her daughter take over the company, and from 2023 we have another executive director who is operating the company. The company is, however, still owned by the same family, and we have roughly 240 employees. So what we do? In Europe, there are two companies which are producing specific pathogen-free eggs for vaccine production. There is a German company and us. We are doing it as a contract manufacturer for a US company, though. Besides that, the other major profile what we are focusing on, those are clinical studies for veterinary development. Here we have the capabilities to work with large animals up to bovine scale, and we also have biosafety 3 laboratories. Many challenge models and many highly experienced colleagues are working there. Of course, to provide a full service, we have dedicated QC laboratories and diagnostic laboratories, what are serving partly our clinical activities, partly our production and development activities as well. We are just completing the renewal of our production plant. This is a small production plant, what I will introduce to you a little bit more details. And from May, we expect to have a full R&D laboratory. Tamás will speak more about that one. Since we are working with eggs, which are quite delicate material, we have our own distributor company, which is delivering our goods all over Europe, from Scotland to Bulgaria. And of course, this is a specific service that we can offer. So let's see our sites. As I mentioned you, the headquarters is at Mohács. You can see at the left side. But all the animal facilities, including the SPFX production sites and the clinical development research and service parts, those are placed in rural areas around Mohács in order to be quite far away from potential contamination sources. SPFX, as I mentioned you, that these are produced by very specific poultry genetics. Different genetics of eggs are produced, which are driven by the customers. We produce the eggs at 3 sites. Those are tested according to the European Pharmacopoeia or if needed by any other tests. We supply these for veterinary services or veterinary producers, but also for human producers all over Europe, Spain, Germany, France, Scotland, as I mentioned you, just to mention Italy, just to mention some. Here we have the capability producing IGY as well. However, this business is not working at the moment, so we are not producing IGY, but we have the capability to do it. When we speak about SPFX, of course, we can supply embryonated eggs to different ages as well. I mentioned you that besides that we have laboratories which are capable to make all the relevant measurements what are needed for the clinical studies. So these include microbiological laboratories, serological laboratories, tissue and virology laboratory, and of course molecular biology. The clinical services and these laboratories are running under GLP. From tomorrow until Thursday we are going to have a GLP inspection. Hopefully we will pass it again as since 2006 every time. Besides that, of course, besides measuring the clinical material and giving as a service for clinical tests, we are capable to develop and validate analytical methods as well as challenge models or clinical challenge models, and we have some in-house challenge models as well. Some words about our manufacturing organization. This is under my lead, by the way. Here we have a small laboratory which is capable to produce, or we try to focus on virus-based products, inactivated or live. So in case of live viruses, we can freeze the products mostly. And we have different partners who can help us on in case of the inactivated vaccines for the formulation and filling. Now, we have two different businesses. One of it is to produce our own vaccines, and the other it is to make toll manufacturing. We hope we are going to have the GMP qualifications from April. Currently, we are finishing the qualifications of the laboratories, which went over a quite severe refurbishment in order to meet with the renewed Annex 1 of the GMP requirements. As I mentioned you again, we can do egg, otter and cell-based or productions mostly, but of course any other funky procedures that you have, we are happy to try. We have some potential cooperating partners who ask things what I cannot tell here, but they have quite new approach to productions. And as the other directions, what we are doing, we have our own product or products. Here I show you one on the picture, which is an inactivated virus for goose or geese. This is quite specific. Actually, so far unique in the world, I believe. And I will ask Tamás to tell a little bit more about this product and also on our R&D facilities.
Thank you, Bálint. Ladies and gentlemen, my name is Tamás Cömpő. I'm head of the newly established Research and Development Department at Profil. What Bálint talked about is already in existence. What I talk about is just a plan and under construction. So basically half a year ago, the owners decided to further improve Profil with the establishment of an in-house research and development lab. And this lab will focus on providing R&D services in protein expression, immune analytical method development, and vaccine developments. But more importantly, this will serve as the basis of Profil's future developments, future vaccine developments for its own portfolio. Bálint already showed to you the only and single existing vaccine which we have as of today, that is for a goose hemorrhagic disease. But our plan is to build a little portfolio in this niche market. The construction of this new laboratory will be completed by the end of May this year, and we already recruited the staff, and we plan to have competencies in recombinant DNA technology, qPCR, or any other hybridization-based assay development. We will capable to express recombinant proteins on all 3 major systems. We can develop antibodies, monoclonal, polyclonal, to any given target, and build those into immunological assay platforms like ELISA, starting from ELISA, but going through complex microbead-based assays, or to even field tests as required. And by combining these competencies, we hope that we will be able to provide a CDMO-like service, building on the other legs of the company. If we develop something here, that could be tested in our animal facilities, and even the manufacturing technology could be developed, and test productions could be run even under GMP if required. So since this is a scientific conference, we thought to give a little example of what we have done so far in the last couple of months with our R&D lab and with our proper staff. As we mentioned, we have a vaccine, an autogenous vaccine against a hemorrhagic polyomavirus of the goose. This causes hemorrhagic enteritis and nephritis in goslings with nearly 100% mortality. And I'm sure you are not familiar with the goose production, as I was not familiar, I don't know, 1 year ago. But there are 2 kinds of goose. One is the gray variety, which is used for liver production, and the other one is the white genetic variety, which is mainly used for meat production. And this virus so far caused issues only in the gray variety. It was present in the white one, but caused no clinical symptoms and disease. But in October last year, it started to cause issues here and there in the region. So we isolated the virus, sequenced it, and found it to be different in 1 amino acids, in one of the anchor proteins of the outer structural protein, which could be potential explanation or not. We will see when we do the controlled animal experiments in our facilities. This example just shows that even small things, and if we keep our eye on the field, we can find something, and that might be scientifically interesting, and at the same time, if we can translate it to our vaccine market, it could be also potentially useful commercially. So thank you for your attention. Our vision is great. We want to develop Profil to one of the region's dominating company in human and animal services by combining our unique competencies in SPFX production, the huge experience in preclinical and clinical animal experiments, manufacturing, and R&D. Thank you.
Okay. At the end of this section, please, Bence Téglás from the Target as Kft. Not Kft. Company, I'm sorry. Please take your presentation.
So hello, good evening, everyone. I'm Bence Téglás, Sales Marketing and Business Development Manager of TargetX Biosciences Hungary, and I'm here to represent my firm. And of course, I would like to just highlight our portfolio a little bit and a little bit of the cooperation of what we did over the past 2 years with VNL here. So yeah, here we go. So there are a few disclaimers here. So since, as it is written, I'm a sales, marketing, and business development manager, but disclaimer number one is that I'm also a scientist, so we can also talk in the same language. So I'm not just a sales talking person. The other thing is that I have sent out this presentation on my way to my annual skiing trip, and there must have been like a speed bump or something over the Slovenian motorway. So I said I actually sent the 1.3 version of it and not the 1.4 version, so there might be some spelling errors there. Other than that, yeah, let's just start with a short introduction. So our very basis of our foundation is a very knowledgeable and experienced staff, and of course the inspiring and productive atmosphere that is generally present all over TargetX. And we really believe that the rock-solid base of scientific innovation are these two key pillars. Our main portfolio actually covers products and services at the same time. Of course, high-quality biotech services usually cover protein engineering, vaccine development, and VLP-like vaccine development platforms. I'm going to talk about virus-like particles in a minute. There is also protein characterization services, so if you have an existing protein and you might want to understand how it works, how it actually contributes with other either proteins or other small molecules, then of course we can do that for you. And there is also drug discovery support, which is still in our portfolio. As for the products that we produce, there are like protein components for diagnostics, such as in bulk formats, like in larger quantities. We also have end-user products, so I'm also going to talk about one of these, and we also have diagnostic kits in our portfolio. So in general, it's all about proteins, product development in terms of proteins, of course, ISO certified production as well, custom biotechnology services in the field of protein biotechnology. And of course, we are also moving on to constant innovation. This is one of the basic drives that we have in the company. But what is our history with VNL in general? So I'm only going to talk about the past 2 years. First of which was actually a virus inactivation portfolio. So the thing is that this whole project was executed on behalf of a large pharmaceutical company, so therefore there is a non-disclosure agreement, so I can't really show the details, but the main idea is described here. So yes, we actually had to produce VLP in high purity and in large scale. However, the VLP protein was expressed in an insect cell, actually high-5 cell, transfected by a baculovirus. So after the first steps of purification, baculovirus resistance residues might still be active in the purified product, so we definitely needed to inactivate the virus without destroying the delicate structure of a virus-like particle. So, Roland, do you have any idea how to do that? Exactly, a few slides ago, we actually just heard that exactly. So UV inactivation was actually the perfect solution to do that. So that's where we started our collaboration with VNL, in particular Rolling, and of course Roland and Daniel just actually followed the whole project. So this was our first cooperation here. The other thing is that we also have an RNase inhibitor, and of course, since there are RNA-based viruses, we just wanted to show some samples to VNL. Of course, this is a very specific kind of product because it is also expressed in insect cells. Therefore, the protein itself has way better folding properties, comparing it to like bacterial expression, and of course, it's endotoxin-free. And it is extremely reliable, meaning that it can also tolerate over 30 minutes in 65 degrees Celsius, and of course high purity, and it can inhibit all A, B, and C RNase classes. And after sending out the samples to VNL, of course there were some in-house tests. I have just discovered today how the in-house test went on, so it was thoroughly tested. And after all of that, of course, we just signed a contract, and there is still an ongoing partnership over this product. And finally, I also wanted to highlight, just one extra thing to mention. I still have samples here, so if you want to try out this product, if you're interested in RNA work, I have 5 samples in the freezer, so I can give it out to you. And yes, over the past year, we are very proud of this project. It's an ASF-free diagnostic qPCR kit that we developed over 10 months. ASF-free stands for African swine fever virus, and it's a triplex qPCR kit with all relevant controls involved. It is validated by the EURL, which is basically the European Reference Laboratory of this particular pathogen. Therefore, this is basically the highest quality standard here in Europe. And our product seemed to be like 20 to 30% quicker than all the competitors. And yes, we actually had some reliable scientific consultation with VNL here. This was a huge work basically. So we did the whole development of the product itself. We did the testing, we did the validation with the Madrid laboratory. We also had the equipment suitability tests, ISO certification, all the marketing, packaging, everything within 10 months. So yeah, we are very proud of this product. This is going to be one of the flagship products of our company. So yeah. Yeah, it is ISO certified, the kit is ready for shipping, so it is also a great product. And of course, VNL contribution and collaboration was also part of this success. As you can see, this is basically a part of the validation procedure, a few figures here. So as you can see, the different curves pretty much align. And here we have a diagnostic sensitivity of 98.5% and diagnostic specificity of 100% based on the reference laboratories testing. So yeah, we are very proud of that. And yes, why are we here today? As Gabor Kemenesi earlier mentioned, of course, we are here to celebrate, but we are also here to do some networking with partnering with potential partners here in Hungary. So I just wanted to highlight the key basis of partnership that we figured. And this is basically reliability, flexibility, and sophistication over our projects. So we are here to do some networking as well. So we hope that most of you are also going to show up at the later wine tasting. And I'm not gonna, I know that, that everyone is kind of tired as I'm a last presenter today. So thank you very much indeed for your patience, and thank you very much.
Yes, as Bence said, we reached the end of the program for this day. I thank everybody who are here, and thank you for your the presentations, the presenters, and I hope everybody joined this day, and have a good rest today, and see you tomorrow. Okay, so I close this day.